My name is Simon Young. I'm Senior Programme Manager for Dementias Platform UK, and I'd like to welcome you all to our third conference, our third translation conference. Looking forward to discussions across the three different themes that we have in the conference, being biomarkers, data, and trials delivery. Great to see so many people here at the conference. I'd like to thank particularly our sponsors, Alamar Biosciences, AD Data Initiative, Alzheimer's Research UK, Alzheimer's Society, ASITE in conjunction with Biogen, Lilly, Novo Nordisk and Roche, which incorporates both Roche Diagnostics and Roche Pharma. I'd like to thank the speakers that have contributed and agreed to speak to such interesting topics that we've got in our programme. And I'd like to thank the ECRs for the posters that they've put up in the room. A lot of interest in that this morning. And the flash talks, some of them will be giving. Please visit the posters and have a look at the interesting science that the ECRs are working on. And please also vote for which poster you feel is the best, because we will be giving some prizes out to ECRs later on in the conference. And in there and also outside, there are some stands from sponsors and others. So please go and have a look at those stands. And that includes the DPUK stand, where you can see demos and you can get literature about the DPUK programme. And in addition to the sponsors, we have stands from Brains for Dementia Research and Joint Dementia Research. And please use social media as well. We're across various social media channels. And we've got a hashtag translation25, so please quote that in any social media that you take part in. With which, I'd like to move on to our first talk. I'm very pleased to introduce Professor John Gallacher for our plenary session this afternoon. John, as many of you already know, is Professor of Cognitive Health at Oxford University Department of Psychiatry and has been Director of Dementias Platform UK for the last 11 years. John has over 40 years' experience in psychology and epidemiology. His research interests include neurodegeneration, aging, cognition and lifestyle. And John is also Director of the Brainwaves Cohort Study and Executive Chair of the MND Translation Accelerator. So with which, I'd like to welcome John to the stage.
First of all, as I'm going to cover a lot of ground fairly rapidly. But I want to say that really I'm standing here on the shoulders of scientific giants. I'm not one for lots of names and photographs and attributions because I prefer to talk about the issues. But nevertheless, the DPUK team is a really great team to work with. And I'd just like to talk about to name check the key scientists in this. And they have contributed to all of it. So there's James Rowe and Atticus Hainsworth, John O'Brien, Simon Thompson, Emma Squires, Vanessa Raymond, Ivan Koychev, Ronan Lyons, and Sarah Bauermeister. Okay? All I'm going to say today is really due to their really high quality work and their dedication. So what I really want to start with is welcome to DPUK. And in one sense, you might think that's obvious, but it goes more than just, it goes deeper than a slide. We really do want to work with you. We really do welcome your input and to our programs and the input that we can make to your programs. So I have a question for you. What are you really good at? Seriously. What are you really good at? Because we want to help you be good at what you're really good at. Okay? And if we can do that within the remit of our funding and our program, that would be our pleasure. And if over the next 2 days there are ideas that you have, thoughts that you would like to pursue, then we invite you to come and discuss them with us. When it comes down to the hard reality of what's possible, but the will is there, and we'd very much like you to partner with us. So Okay. Very briefly, DPUK over the last decade or so has become a global partnership. We've leveraged over £100 million for dementia research over and above our core MRC funding. We have 9 industry partners, 14 academic partners, 3 philanthropic partners. We have a 7-site PET MR network, which you're going to hear about later on, and we relate to 3 international data platforms. And we are absolutely delighted that we've been able to foster those relationships. And the reason we've been able to foster those relationships is what we prize very highly is our high trust, pre-competitive environment. You know, if you want to work with us, we want to be very transparent before you. We want to be very clear about what's possible, what's not possible. And we want to be able to share the risks as well as sharing the benefits. And I think, you know, as we foster those relationships, then the work accelerates and, you know, the trust builds up. One thing leads to another, leads to another, leads to another, and then you have an ongoing program. And our program really comprises 3 components. There's the data portal, our trials delivery framework, and our experimental medicine incubator. Let's just look at our data. We really do offer to everybody in this room very mature informatics solutions. Over the last few years, we've received over 2,000 data access requests from 1,200 users in 43 countries across the world. We have curated, or we host and curate 85 data sets. We've recently received 135,000 brain images. One of the most wonderful things to me is our data and metadata-led federation. And again, all credit to our Swansea team for developing that. With the advent of AI, we've been looking at the security and governance issues around AI readiness, and I think we're there. I think we've resolved that. We're developing synthetic data so that if you like, the access to the data is much more rapid. And we have community-led research hubs, so focus on MND, traumatic brain injury, neurological tissue, iPSCs. We also have hubs around imaging, and we're developing one for genomics. So it's a very mature informatics solution. In terms of our trial delivery framework, we have this sort of mantra. We really want trials for everyone, trials for all. And we have our Great Minds Register, which has been an opt-in register, which allows you much more detailed phenotyping. A clinical studies register, which is using data collected from cohorts so that it's readily available and it goes back many years. We have a 78-site trial network, a portfolio of 27 studies. And one of our flagships is the FAST study, led by Ivan. And that's really looking at how to rapidly collect biomarkers on large numbers of participants. In terms of experimental medicine, we look beyond amyloid tau and neurofilament light. We focus on vascular health, synaptic function, and neuroimmunology. But really, it's the concept behind the experimental medicine I want to get across this afternoon. We want to co-design with industry. We want to share the risks and share the benefits. We want to validate in humans mechanisms discovered and identified preclinically. Then we want to, if you like, conduct precision experiments so that we're able to work with industry to make rapid go, no-go decisions on the many compounds that you have on the shelves, all right? You really do not want to invest $200 million if plus on a phase 3 trial only to discover what works in a mouse does not work in a man, okay? Or a woman. So it's really important that we're able to get together and work out how best to do that. And we do invite you as biotechs or as pharma to come and discuss with us in a confidential manner, you know, what are the burning mechanistic questions that you have that we can help you with. So let's move on to next generation therapies. One of the beauties of the first generation therapies is not so much that they are marvelously efficacious to vast numbers of people, but actually they inspire industry to develop the next generation, which by definition will be an improvement. They'll be likely to be more effective, they're likely to have a wider range, and there's likely to be greater public requirement that they are funded by people like the NHS, for example. So, where are we up to? Well, as we look for next generation therapies, I think DPUK has to, and it doesn't just do this reluctantly, it needs to, and it is delighted to align with the ongoing investments in the UK. So the government has put up tens of millions of pounds looking for funding to co-fund from industry, and their initiative, the Neurogeneration Initiative, really boils down to basically 3 pillars. Now, some of you will be very familiar with this. To others, this may be news. Basically, there's a biomarker pillar, looking at the development and accessibility and the application of new biomarkers, mostly blood-based, but not exclusively. There's a trials pillar, bringing that biomarker information to use in developing phase 2, phase 3 trials. And then there's the treatment delivery pillar, which is waiting to be developed. Now, there are already substantial investments in these areas. For biomarkers, there's the Dementia Trials Accelerator. That's been funded to the tune of £20 million by UKRI MRC. And then for the biomarkers, then for the trials, you've got the Dementia Trials Network and the Research Development Network funded by NIHR to accelerate the conduct of trials throughout the UK. And what this is about is making the UK a really good place to come and do trials. Sometimes the UK is very well intentioned and it has lots and lots of clever people, but basically the bureaucratic pipeline going from design to governance to ethics to first through the clinic, etc., takes a long time. So what the UK is really focused on doing is making this an efficient place for doing these trials, but not just for doing big phase 3 trials, for doing phase 2 and phase 1A mechanistic studies, and also, as I alluded to earlier, if you like, the observational clinical studies that are required to address no-go questions by pharma. So where does DPUK fit into this? Well, we straddle both biomarkers and trials. As I said earlier, we have a very large trials network, we have trials registers, and we have at least one study, but obviously several also, which are looking at the best biomarkers to use in different trials. So that's where we're going. And what I'm going to do for the next 10 minutes or so is suggest how we might be getting there. And through doing this, I'm inviting you to make contributions and suggestions to us as we do this. Okay, so the future is, okay, that's the keynote. That's the keynote, repetitive message. The future is, we consider it, part of this future is to be providing core data services. You know, data is messy, data is hard, data is complex, data, I should say, are challenging, okay? Well, one of the things that DPUK has genuinely focused on is providing generic solutions to specific problems. And this means our specific solution to this specific problem is very easily adaptable to other problems. And let's take the issue of trusted research environments. They cost a lot of money. They are hard to set up. They require enormous governance services. But if you wanted a trusted research environment, you could come to DPUK and have one within our trusted research environment. So you're really just learning from all that we've done. You're taking all the tools that we've developed, all the safeguard, all the governance that we've developed, and saying, okay, can I have this? It saves time, an enormous amount of time. It saves money, and actually it's low risk because we've taken the risks, we've made the mistakes, and we rectified the mistakes, so we know we can deliver this. Okay, so what we're offering to all of you, industry, academia alike, is we are able, if this helps you, and I think it will help you in terms of time, money, and risk, to provide these core data services so that data can be brought into a secure environment, they can be curated to a common standard, they can be made available via federation to other platforms, and all those other things there which are in the core data services. So if you find that of value, then moving ahead, we would very much like to do that. To demonstrate it, I'd like to direct you to the traumatic brain injury website, TBI Reporter. Basically, if you look at that, it's fantastic. We love it. We absolutely do it. They have ideas and thoughts that we never had. But at the bottom of the page, one of the partners is DPUK. And basically, you've got the whole DPUK framework been adopted by TBI with their own identity, running to their own purposes, with no sense of losing academic freedom. In fact, it's creating academic opportunity for them. Right? And they have really run with it. And it's, we're really, really delighted to see how successful they are. To me, that is an example of what we would offer to the wider dementia community. And then I'd like to talk briefly about federation. Now, everybody talks about federation, and it's very, very difficult. But typically what it means is you federate with me, and I will then help you federate with them, and then I will help you federate with them. So you end up having one, if you like, real source of authority and control. We would much rather have a much more egalitarian system, when if you look at that diagram on the right, where all the platforms have sibling relationships, not paternal or maternal relationships, okay? And what we are doing is offering that to you. So if you would like to engage in that, again, vastly reduced costs, vastly reduced time, vastly reduced risk, then we would be very, very pleased to discuss that with you. So we think the future is providing core data services, and DPUK is very pleased to discuss that with you. We think the future is linking molecular data to health data. There's been a fantastic revolution over the last year or two, I think, of people recognizing that we need to go from clinical diagnosis of pathology to biological diagnosis of pathology. Okay, just take Alzheimer's disease. Is it one disease? Or actually, are there 3, 4 different neuropathological pathways leading to similar outcomes? It's probably the latter. Okay, but what that means is be able to get hold of the proteomics, link it to research cohort data, because that gives you the detailed phenotyping, and then linking that to the outcome data. And we are in the process of discussing with various consortia and with the NHS how we might do this moving forward. So we think it's a very real possibility. And again, we would love to discuss with you if that captures your imagination. Okay. The future is a virtual national trials register with enriched biomarker data. There are many trials registers in the UK, okay? And all of them, and I include ours, are inadequate. There are specialists here, there's specialists there. They have different governance requirements. They have different, if you like, phenotyping requirements. And what we'd like to do is provide the infrastructure, because we've built it effectively, but to apply the infrastructure to bring in these different trials registers together. So we might have a common core, a minimal data set, and then for specialist registers, we might have aligned, more detailed data sets. Now, the core minimal data is actually not too complicated. It just requires people to agree on what to measure, all right? A cognitive test, a blood sample, a family history. It's really not hard to work out what is needed. I think with the aligned data sets, it's more complicated. And I think you do need to work out particular alignments for particular purposes, because it's expensive doing this. And so we have, if you like, a collection of studies under the auspices of DPUK and our collaborators, because we're very interdependent with them, on the right of that slide, saying, okay, these are the things that we can do. Come and help us shape this program, because this program is designed to enable you to have precision recruitment to address a precise mechanistic question, okay, rapidly and relatively inexpensively. because you know who the people are, reduce screen failure rate. All of these things you're very familiar with, but that's really what we would like to do. And finally, the future is integrating technologies for mechanistic studies. The MRC have recently funded an experimental medicine core, and it was overwhelmed in its response, which just shows you the level of interest by UK scientists. But one of the big problems of experimental medicine is that it's very complicated. It's very hard to do. And it's very hard to do because somebody over here is a brilliant sort of imager, somebody over there is a brilliant geneticist, et cetera, et cetera. Somebody elsewhere is a brilliant clinician. And so what we would like to do moving forward is to create critical mass by bringing these technologies together so that it's much easier to get cerebral spinal fluid, it's much easier to get your PET or your MR imaging, it's much easier to get your blood biomarkers done. A sort of one-stop shop, okay? Now we think this would, how can I put this? Deconstipate the experimental medicine pipeline, okay? So if you're interested in that, we'd love to hear from you because we will be putting these ideas to MRC in the next 6 months or so. And finally, I'd just like to acknowledge our partners. We're extremely grateful to them. I think with our programs, with most of our programs, I would say 80% of our programs, we've over-delivered sooner than we promised we would. And the ones that are slightly delayed are due to COVID because you basically stop work for 2 years, then you have to pick up again. So we're in, I think, a good position in terms of delivery. Our partners have been very, very patient with us where we've had to work back because of COVID. And I just would like to express my enormous gratitude to them. So there you are. That's where we're up to, and that's where we're going. Thank you very much.
Thank you. There's many reasons to thank John, and partly for such a fantastic overview of Dementias Platform UK, its past, present and future, but more particularly his leadership over the last few years building this high trust and pre-competitive environment, and that really is very special internationally. For its efficacy, for its long-term vision, and actually to be part of. And the very fact you're here suggests you are interested in that, and we can move forward together. Welcome also to all of you. We have people here from academic, business, industry, and clinical backgrounds. So as well as meeting up with old friends, do remember to talk to someone you don't know. It's a really valuable part of the work of Dementias Platform UK that it crosses all sectors, the academic and the business, the clinical, and indeed the patient advocacy. Welcome to this afternoon session. We're going to focus on the use of biomarkers in experimental medicine. And the reason for that is that although we're all aware of the urgent need for new treatments and better diagnostics for people with or at risk of dementia, perhaps it's not so clear to everybody that the real pinch point in the development of such therapeutics is in this early phase clinical trials coming out of the lab, out of animal models and into the human. It's the point a bit where the risks go up, costs go up, success rates plummet, and there's a major shift in the biology that needs to be addressed as we move from preclinical models into humans. So this early human studies create a pinch point or a bottleneck, and to open up that and to promote the development of new drugs and new treatment opportunities needs experimental medicine. And experimental medicine is not just about having the drug. What is the drug, or what is your drug of choice? Experimental medicine is really about having the tools to study such drugs, and many of them may be coming through, and to do so effectively, to put the right patients into the right studies, have the right outcome measures. All of this involves the work of experimental medicine. A lot of this will revolve around the use of biomarkers. Now, the word biomarkers is very, very well used. Always ask someone talking about biomarkers to put an adjective to it. Is it a diagnostic biomarker they're talking about, a prognostic biomarker? Is it a monitoring biomarker? Okay. Because the needs and the way in which one would benchmark and validate biomarkers depend on its purpose, that context of use that the FDA would emphasise. And I think that's not quite emphasised enough in many discussions of biomarkers. Put an adjective to it. Okay, make sure that you and the person you're discussing with know what type of biomarker you're talking about. And then we can move forward, as indeed we'll see this afternoon. We'll have some discussions around imaging biomarkers, blood-based biomarkers, digital biomarkers, individually, and how they can be understood jointly. So the UK is a particularly exciting time at the moment. I know colleagues from the States are having a number of challenges at the moment, and our hearts and sort of sympathy go out to them, and I do hope things settle, especially for the earlier career researchers in the States, very uncertain times. In the UK, we're very fortunate. There's still a period of very strong political and strategic interest in dementia, and the need is being recognised with large-scale government investment, particularly through the Dame Barbara Windsor Dementia Mission, as well as now the Dementia Goals programme. And that's fuelling investment and co-investment in dementia research through things like the Dementia Trials Accelerator, the Neurodegeneration Initiative, Dementia's Platform UK, and indeed ongoing a very large investment from MRC in experimental medicine with nearly £20 million on the table through academic co-investment with industry partners for experimental medicine focused on dementia and degeneration. So we can expect these to really accelerate the UK field, developing capacity, some really exciting science. But to encourage us along the way, we're going to have this afternoon session to learn from real-world experts to tell us how it's really done, how should we be undertaking and using biomarkers to promote experimental medicine. And the first of our speakers this afternoon, I'm very glad to see, has made it over across the water. So Donna Wilcock, going to welcome Donna up to the stage to come forward. Donna is from Indiana University School of Medicine. She's the director of the Center for Neurodegenerative Disorders there, the Barbara and Larry Sharp Professor of Alzheimer's Disease Research. And her research is extremely diverse, but she's probably best known for this work linking vascular and Alzheimer's pathologies. As part of a sort of common pathogenic understanding, and never has that been more important than now that we have amyloid-based therapies constrained perhaps principally by their vascular side effects, and how are we going to get past this barrier? So Donna, very welcome. I'm going to hover over there to steer questions after your talk. There'll be some pressing questions after your presentation, and then a general discussion later on in the afternoon. Donna, welcome.
Thank you all so much for that welcome. Thank you, Professor Rowe, and thank you to the organizers for giving me a 2-day mental break from our current political situation. It's definitely a tumultuous time. We were talking about it over lunch, but we are fortunate, I think, that in the long run, dementia has historically been a bipartisan issue, and I hope that it will continue to be. And so when the dust settles, I think our research will continue to grow. If not, you'll see me moving back to the UK, I think. I am originally from Sheffield, but you wouldn't know, it's been 25 years in the States. So what I'm going to talk about is kind of give an overview of where we are with fluid biomarkers, in particular plasma biomarkers, not just with Alzheimer's, but across the dementia spectrum. And that's really because of what the real picture of dementia is. If we want to move towards a precision medicine type approach to prevent and treat dementia, we are going to have therapies and biomarkers for all of these different dementia-causing pathologies. You heard about Lewy body earlier from Professor O'Brien, who showed a very simple pie chart, which I think as academic, at least when I was in graduate school, we all saw that pie chart, and it was very clean. 50% are Alzheimer's and 25% are Lewy body. And the real picture that emerges across now many hundreds of autopsy studies is that there is so much significant overlap of these pathologies when you look at the brain of someone who suffered from dementia. So we do have a large swath that have Alzheimer's pathology, plaques and tangles, but we also have another very large swath with significant overlap of vascular contributions to dementia. That includes microinfarcts, microhemorrhages, cerebral hypoperfusion. Typically, we relate that to white matter changes in the brain. But we also know now that there is a significant amount of TDP-43 proteinopathy that can produce late limbic predominant age-related TDP-43 encephalopathy. I'm only going to say that once. And then obviously we have the Lewy body pathology, but in a vast number of cases, we see some levels of plaques and tangles, plus a few Lewy bodies, plus some TDP-43, plus some microinfarcts and arteriolosclerosis. And so this is just another figure taken from a former colleague and still friend of mine, Dr. Pete Nelson at the University of Kentucky, who showed that with increasing age, yes, autopsy-confirmed Alzheimer's disease increases, but essentially peaks around 90 to 95, and then you actually see somewhat of a decrease in Alzheimer's after that age, whereas if you look at hippocampal sclerosis, which is the old term for what we now call late, that starts to really increase once you get past the 75, 80-year time point. And of course, cerebrovascular disease past the 80 years of age, you really see a lot of cerebrovascular pathology. The line drawn here is just showing the average age of a very famous, heavily referenced paper that was used to predict what Alzheimer's prevalence, how Alzheimer's prevalence would increase with age. But the average age of their study was actually 75, and we know that people are living much longer now. As people live much longer, we see a lot more of these co-pathologies occurring. So when we think about those pathologies, we really need to think about biomarkers for all of those pathologies. I would say we've made by far the greatest progress in Alzheimer's biomarkers. So for tau pathology and tangles, we know CSF tau, emerging CSF and plasma biomarkers, but also now emerging data that some of the plasma p-tau markers that we've used for amyloid deposition are also actually heavily related as well to the tau burden in the brain. And then, of course, in addition to the fluid biomarkers, we have tau PET imaging available to us now as well. With respect to the plaques, we've had for quite a number of years now CSF A-beta 42 to 40 being kind of a gold standard biomarker that's been used. And then in more recent times, I would say, the last kind of 3 to 5 years, there's been this huge acceleration in our ability to measure some of these proteins in plasma, thanks in part to technology and in part to, I think, ingenuity and innovation of our scientists. Plasma p-tau 217, I'll talk about some of that data, p-tau 181, and also plasma A-beta 42 to 40 ratio. And then, of course, we've had the amyloid PET around for a number of years now. I would argue that we would not have the anti-amyloid therapies that are approved now without having this arsenal of biomarkers. Before we had PET imaging, before we had the CSF biomarkers, when we ran trials in Alzheimer's disease, we really were just feeling our way around in the dark. We were relying on clinical diagnosis, which we know even for the absolute best clinician is only accurate around 75% of the time. And that number is usually closer to 60%. A lot of our early trials that we say failed may not have been failed drugs, but instead in the trials, we didn't have a very clean Alzheimer population, and we probably had people with other dementias in those studies. But we really don't have any biomarkers for TDP-43 right now. We have some for alpha-synuclein. You heard from Professor O'Brien earlier about the dopamine imaging for Lewy body dementia. I'm going to talk today about fluid biomarkers, so I'm going to talk about the RT-QuIC assay that I think is really moving this field forward. And then for small vessel disease, which is the most common vascular contribution to cognitive impairment and dementia, we've heavily relied to date on the presence of white matter hyperintensities in the brain by MRI. But now there's so much data telling us that there are other contributors to WMH, including amyloid deposition actually seems to contribute to the presence of some of these WMH. And also we think the presence of WMH is kind of the tip of the iceberg, and there's a lot of damage that's happening to vessels before you start to see these signals on MRI. So there is a push to try and identify more sensitive biomarkers for vascular injury as well. So I'm going to start with plasma biomarkers of amyloid and tau, which are now moving into the clinic. We have studies in the US, and I'm assuming here in the UK, trialing these plasma biomarkers in primary care settings, and especially in people who have memory complaints and may potentially be eligible for those disease-modifying therapies. So the next two slides are taken from the same publication. I really like this study just because it's some of the largest fluid biomarker groups. It's the BioFinder 2 study and the NIGHT ADRC that came together, and they compared the sensitivity and specificity of plasma, in this case, percent p-tau217, so they're looking at the percent of 217 that's phosphorylated. They also had CSF p-tau181 to the A-beta42 ratio, and then they had CSF A-beta42 to 40 ratio, which, as I said, had been kind of the gold standard for a long time for a fluid biomarker. And so what you can see is, and this is all relating to amyloid PET positivity, is that the plasma percent p-tau217 actually outperforms those CSF biomarkers, and that is in all individuals, but they broke this down further into the cognitively impaired individuals and the cognitively unimpaired individuals. So this is the BioFinder study, and this is the NIGHT ADRC study. You can see there's fairly comparable numbers across those two studies for these biomarkers. And so plasma percent p-tau217 and the CSF p-tau181 A-beta42 give almost identical area under the curve, so almost identical sensitivity for both cognitively impaired and cognitively unimpaired. And when you compare the plasma and CSF biomarkers, the plasma actually seems to outperform those gold standard CSF biomarkers in the cognitively unimpaired groups, which suggests that maybe it's more sensitive for people who are in the preclinical early stages of disease. And so this is amyloid PET. In the same study, they also looked at the sensitivity and specificity of these biomarkers for tau. And so this is tau PET positivity. You can see that generally these numbers fall a little bit lower than the amyloid. But especially in the Biofinder study, you can see that the plasma percent p-tau217 for all individuals, you're still looking at around a 0.95. It's more sensitive. It seems to be the area under the curve is higher in the cognitively impaired individuals as opposed to unimpaired individuals. Again, similar numbers in the night ADRC cohort as well. As you would expect, the CSF A beta 42 to 40 really doesn't do much to predict brain tau. But this percent p-tau217 in the plasma seems to be very sensitive for brain tau burden as well as brain amyloid burden. And so including a p-tau marker in the CSF as well as A beta increases the sensitivity to tau. So if you're doing a CSF collection and you're relying on CSF for your biomarkers, if you can include a p-tau, it will give you information about tau burden as well as amyloid burden. This is just some data from our own ADRC. In this case, we're looking at p-tau217, not percent. So this is the Quanterix, actually the AltPath p-tau217 assay that's run on the Quanterix platform. And what you can see is we actually instead of assigning a single cutoff, we have established a negative cut point, a positive cut point, and then we have an intermediate, kind of indeterminate group. And so we had 52 individuals who were amyloid negative and 48 individuals who were amyloid positive down here. And what you can see is that the ones that were p-tau positive, the vast majority of those fall into the amyloid positive. We had two false positives, so this read as positive, but in fact was amyloid negative on PET. And then when we look at the p-tau negative, so this is below the 0.4 picogram per mil cutoff, we actually had 6 that were falsely called negative. So they were amyloid positive on PET, but the plasma levels were negative. And we had 14 individuals, who fell into this intermediate category. The thought that, so our population is, in Indiana, is very much a community-based population. We have a lot of health comorbidities in our population, including diabetes, hypertension, hyperlipidemia. I don't want to, well, they're fairly unhealthy. And so we're exploring, is some of these comorbidities potentially chronic kidney disease, which we know can affect the readout of some of these biomarkers, is that the reason why we saw some differences. So overall, we saw a negative predictive value of 88 and a positive predictive value of 95. So this is interesting. This is now, I think, the field has kind of coalesced around p-tau217 being the plasma biomarker that we should be using. It seems to have a slightly higher sensitivity than the 181. It seems to be less sensitive to some of those health comorbidities that can affect the concentration values that you're reading. So one question that I think has hovered over us is how do these plasma biomarkers change with therapy? We know that if we give an anti-amyloid therapy, we can see the amyloid being cleared on PET, but can we also track that using plasma? Obviously, from a real-world standpoint, if we didn't have to do amyloid PET on everybody, the cost to treat people would be significantly reduced. So this is changes in plasma levels with donanemab treatment. And what you can see is the gray line is the placebo group for p-tau217, and in this case, plasma GFAP. And then these are the donanemab individuals in green. This is separated by the dashed line are the individuals who stopped treatment at 24 weeks because they were deemed amyloid negative. And the solid green line are the individuals who continued treatment after the 24 weeks because they were still positive on amyloid PET scans. And you can see that the p-tau217 actually does track quite well with what was seen in the amyloid PET scans in this trial. And the ones that stopped at 24 weeks do seem to show that reduction a midpoint compared to the ones that continued on therapy. But what's interesting is that we know that the amyloid remains low even though treatment is stopped at 24 weeks. And the p-tau217 also seems to remain low after treatment is stopped. So this does seem to provide us with a good readout of how a person is responding to these anti-amyloid therapies. I think it's interesting if you look at the GFAP that, and you're always careful to overinterpret data like this, but it does seem as though the group that stopped at 24 weeks, that the GFAP kind of has this rebound back up by the end of the clinical trial. So moving on, I think that we still have a lot to learn about the plasma biomarkers for AD, but this is really a very mature area now, and I think we're going to see in the coming, hopefully, months, I think within the year, some appropriate use guidelines for how do we use these biomarkers, when do we use them, should we be doing p-tau217 on everybody in primary care, who are 50 and older? Probably not, especially not at this stage. The therapies are only approved for people who have mild cognitive impairment. And so if someone was p-tau217 positive but had no cognitive impairment, you're just really worrying them and there's no treatment option. I think if the prevention trials like the AHEAD 345 trial, I think if they end out reading out positive for prevention, we're going to see a different use of these biomarkers in the clinic. So into what is more unknown, which are the biomarkers for those other pathologies. And I really think that what we've learned from the AD field, we need biomarkers in order to be able to do good clinical trials with good readouts and get a positive outcome. That really does prioritize identifying biomarkers for these other pathologies so that we can do the right trial with the right drug at the right time. So for VCID, there's been a number of proposed biomarkers, PDGFR beta, neurofilament light. which, NFL seems to go up, I think, with everything that causes dementia. But there's some evidence that maybe NFL and GFAP as a ratio may indicate vascular damage. So there's some discussion around that. There's some data on using exosomes and placental growth factor, which is what I'm going to talk about. So I've been a part of MarkVCID, which is a NIH-funded consortium within the US to try and identify more sensitive and specific biomarkers for small vessel disease. One biomarker that came out of that so far is plasma placental growth factor. It's a member of the VEGF family. It's named for where it was identified, so the name is confusing when you talk about placenta and old people, but it's simply just the naming of the molecule. So this was the study that was published by MarkVCID. Jason Hinman is the first author on this. And what you can see is if you use Fazika's score, and the CDR for cognition, or if you use the UDS-3 executive function score and the volume of white matter hyperintensity, we see that the diagnostic accuracy for plasma PLGF is 0.89 for vascular dementia. And for vascular cognitive impairment, so these are people who are MCI, not dement, the AUC was 0.74. If we use what is probably a more sensitive measure, which is the volume of white matter hyperintensities, we see that the presence of just small vessel disease is 0.73. If we add in cognitive impairment, we get 0.78, and then dementia 0.85. Again, they're not the 0.98 numbers we see with p-tau217. There's room for improvement, but we're exploring that in the form of different angiogenic factors and potentially building in a ratio of PLGF with something else. But we asked the question of, is PLGF actually in the brain? What is it doing? We do see that individuals, so these are autopsy human tissue from people with severe small vessel disease on the right, or age-matched control with no evidence of small vessel disease. We do see an increase in PLGF labeling, primarily around the blood vessels. And in these same individuals, if we looked at their plasma or the gene expression or the brain protein expression, PLGF was up in all of those. So this effect does seem to be real and to be related to injury of small vessels in the brain. So RT-QuIC, this is an emerging fluid biomarker. I don't want to say emerging, it's been around a while, but I think it's starting to be more widely used for the detection of alpha-synuclein pathology in individuals who have a questionable diagnosis. This is just showing you how the RT-QuIC assay works. For those of you who may not be familiar, cast your minds back to maybe undergraduate or high school biology, to what a PCR looks like for DNA. So you take the patient sample, and if the patient sample has abnormal alpha-synuclein in it, and you combine that with purified alpha-synuclein protein as a substrate, if you, you have your fibril, and then you basically go through temperature changes and you shake to disaggregate the fibril. If you have abnormal alpha-synuclein to start with, you're going to keep amplifying that alpha-synuclein. It's going to imprint basically on the normal synuclein. So you'll get more and more and more fibrils with each cycle, much like PCR. And the readout is a fluorescence intensity. So we can do this on skin biopsy. We can do it on cerebral spinal fluid. It's not. People have tried plasma. I've seen mixed data on plasma, to be honest. Some people have published it works great. Other people have shown that it does nothing, and we haven't tried it, so I can't tell you what the reality is. I think the field will work this out. But this is just showing you how sensitive it is. So in this paper, they had individuals with Parkinson's and Lewy body dementia, and then they had normals, and then a series of different neurodegenerative disorders going across the x-axis here. And then what you see is the fluorescent readout, basically, of that RT-QuIC assay. And so you can see it's very sensitive for the alpha-synucleinopathies. We really don't see any signal across these different conditions. But what I'll say is, yes, it's very specific and sensitive, but it doesn't give you really any indication of severity of disease. So it doesn't tell you how much alpha-synuclein pathology somebody may have. It simply tells you whether they have it or not. It's better than we've had, but we do need to get to a more quantitative biomarker, I think, if we're really going to say whether someone's dementia is predominantly Alzheimer's or predominantly Lewy body. So TDP-43, proteinopathy, there's been a couple of very interesting papers come out in the last few years that I think give us some promising directions for TDP-43 biomarkers. The first one was by Rob Rissman, who showed that astrocyte-derived exosomes in the CSF, this was cerebral spinal fluid. If he isolated those astrocyte-derived exosomes from the CSF, from cases of late, autopsy-confirmed late, and then had non-TDP-43 dementia cases as well as age-matched controls, those astrocyte-derived extracellular vesicles had significantly elevated TDP-43 protein compared to astrocyte-derived exosomes from non-late cases. And so a challenge with this, I think, is the same challenge with any exosome-based potential biomarker, extracellular vesicles and exosomes. There's challenges with scalability and reproducibility, especially if we start thinking about really using it for clinical applications the way we're thinking with p-tau217 right now. This was a very exciting one for me, which is that when TDP-43 becomes dysfunctional, cells will start producing what are called cryptic peptides, because the role of TDP-43 is to regulate that protein translation. And so when TDP-43 isn't working because it's in an aggregate, you now get the formation of these cryptic peptides. And so this paper from last year by Irwin and colleagues showed that there was this particular cryptic peptide that they could measure in the CSF of both familial and sporadic ALS patients, which involved TDP-43. They did not do late, they did not do dementia, so this is very early stage work, but I do think it's a promising avenue. There are a number of cryptic peptides that are known to be produced when TDP-43 becomes dysfunctional, and so I do know that there's a number of groups now that are trying to look at cryptic peptides as potential biomarkers for TDP-43. So this is my last slide, which is for all of you early career investigators, how do we advance our ADRD biomarker discoveries? So how can you come up with your own research ideas that could move this field forward. I think autopsy studies with matched antemortem fluids are really the gold standard that we should be using for our biomarkers. But I think mouse models and iPSCs can help us identify candidate biomarkers that might match specific subtypes, especially when we're talking about biomarkers of inflammation, where there's so many different states of inflammation and beneficial and detrimental, and which ones do we want to increase and which ones do we want to decrease. So I think iPSC studies and mouse models can really help with that. We need ground truth for ADRDs. And so again, while ideally this would be autopsies, we can't really achieve the big sample size we need just with autopsies. So I think we really need to have movement in the area of PET ligands for alpha-synuclein and for TDP-43 so that we can have that ground truth that then we can develop our fluid biomarkers around. And I think as we get improved technologies to optimize the isolation of those brain-derived exosome approaches, I think that will broaden the biomarker potential of using exosomes. So the more we could automate that exosome process, I think the more reliable they could become as biomarkers. So with that, I'll thank everybody in my lab and my colleagues and collaborators and my funding sources for now. And happy to take, I don't know how I'm doing on time, questions maybe. Thank you for your attention.
Thanks for the very nice talk. I'm interested about your landing slide, the idea of the core pathologies. Try to get your thinking around how those come to be core pathologies. So if we incorporate the dimension of time here, do you think there are specific sequences of core pathologies and specific trajectories in which if you just give it time, you will end up having some of them, but not some others? And whether in a fantastic ideal world in which we have perfect biomarkers for every one of them, longitudinal studies will perhaps allow us to know that.
Yeah, so I think you're asking a question that right now we don't know the answer to, which is, kind of a which comes first, right? Does the amyloid always come first? Or do Lewy bodies come first in some people and then the amyloid comes? And then, or does vascular come first? There's some evidence now, even in those familial early onset AD cases that some vascular markers may precede, the detection of amyloid. And so I think it's a great question. I think biomarkers are going to help us answer that. One study that we did when I was at the University of Kentucky was actually a beautiful study by our epidemiologist, Erin Abner, but it was a kind of a collective study where we found that with the presence of each proteinopathy, so if you just took people who had amyloid and tau at autopsy, the likelihood of them having a dementia diagnosis was modest, right? So a lot of them were either normal or MCI. And then if you layered on alpha-synuclein, they had a higher likelihood of having a dementia diagnosis at death. If you added on to that TDP-43, they had almost all of them were demented if they had all 4 proteinopathies. They didn't include vascular in that analysis because almost everyone over 80 has vascular changes. And so they ended up terming this QMP, quadruple misfolded protein, where if you had all 4, it was almost certain you had dementia. Now, the question would be, obviously it's an autopsy study, right? So those people who only had amyloid and tau, had they continued to live, would they have developed other pathologies? And so, yeah, I think we need longitudinal biomarkers that are accessible, and we need to do those studies because we don't know the answer. We don't know if it's homogeneous or if it's an individual to individual, variability. And I think that understanding will drive therapeutic approaches, right? So if it's always amyloid, then obviously we have a tool to lower amyloid now. But in some individuals, if the amyloid comes a lot later than the TDP-43, then maybe the TDP-43 is the driver, and that's what you need to target therapeutically. So we just don't have the tools to answer the questions that you're asking, but I think that's why it's so important. We need to develop these biomarkers.
Thanks for a great talk and
Thank you.
a nice overview. Can I just ask about the using PET as an outcome measure? I can understand for selecting people for clinical trials where you're targeting amyloid, that's helpful. In clinical work where we know that people will have positive amyloid PET but not necessarily have symptoms, is there a role for having sort of clinical outcomes alongside PET?
Yeah, I think this is an evolving landscape. I think cognitively unimpaired individuals who, for some reason, have had an amyloid PET and it's shown positive, maybe that warrants closer monitoring of those individuals. As I said, when we were talking about the use of the AD biomarkers, I think the trials that are happening right now around prevention are going to drive that discussion about when it is and isn't appropriate to use these biomarkers, including PET. I think with lecanemab approval, we really don't have any, in the appropriate use guidelines, we really don't have any need to do an amyloid scan at any point during their treatment. Although I will tell you in the US, some private insurance companies are asking us to do the amyloid PET to prove that the drug is working and that they should keep paying to have the patient on the drug. With donanemab, the way the trial was designed was that if they were amyloid negative 6 months into the trial, then they could stop the drug. And that is what's going to come through in the appropriate use guidelines. I think the recommendation is 12 months with donanemab, and you do a PET at 12 months and then you stop treatment. And then, the question is, do you have to keep repeating the PET to look for the amyloid coming back at some time? The goal would be that, and I think the data is promising, that we would be able to use p-tau217 as a surrogate instead of always running, the amyloid PET, because, as you know, it's costly. And then there's also a lifetime radiation exposure that we could start hitting on at some point with all these PET scans.
Hi. Sorry, this is going to sound like a basic question, and I apologize in advance, but I'd like your opinion.
No basic questions. First rule.
No, I'd like your opinion. Basically, some studies suggest that there is a variability in the accuracy of 217 based on the time of day. And as we move into clinical translation with things like readout in the UK here, do you think, A, why do you think this happens? And B, do you think we need to go to a standardized protocol like fasting bloods we have?
Yeah, so that's an interesting question, right? Because I think a lot of the centers that you're seeing studies from do have standard protocols in place so for our ADRC, we always collect fasting blood the morning of their visit. And so we're not able to see that kind of diurnal variability. I wouldn't be necessarily surprised that time of day or, what you've eaten that day may slightly influence some of these measures, because we know that there are other things. There's some medications, some cardiovascular medications that can alter the levels of these, as well as chronic kidney disease, as well as obesity. Time of day, we know that there's fluctuations in A-beta circulation over the time of day. I'm a little more surprised that there is with p-tau217, because that's thought to be more of a cellular response to the amyloid, which you would think would be a bit more stable than just A-beta production, because we know, like at night, you get higher A beta overnight, and then it's less during the day. So I don't know why we're seeing that. And I think the more we get into the real world, instead of in our very tightly controlled cohorts that we make fast for everything, and the more we're going to learn about all of these little intricacies. And I think we probably will see guidelines along the lines of fasting, taken before maybe 10 or 11 a.m., the way we do with blood glucose right now. Yeah.
Fantastic. Thank you very much indeed. I'm afraid we've run out of time. I'm sure there would be many more questions. There's certainly more work clearly to be done, but thank you very much indeed.
Thank you so much, everyone. Thank you.
Thank you very much, Donna, for very good introduction to the problems of actual imaging with PET, because there are quite a few, and I would like to make it a little bit more understood how difficult it can actually be, and what we do to make it as feasible, and also to understand the PET data whenever you come to work with it. So everybody knows that PET NMR together were used quite successfully in clinical trials in the last 2 recent drugs approved by FDA, and that these were used for stratification. So up front, you would like to know which subjects are most likely to benefit from the trial, but also the response to the therapy. So PET particularly is like computed tomography as well. They are already very well established in oncology. In dementia, we are actually starting this. So it's a very good opportunity to move ourselves a little bit forward. And if I can move forward here. So I would like to show what is actually involved in PET imaging. There are different stages, and what can we make better in those stages? I suppose this one this one works. All those stages from the very beginning of patient recruitment and actually producing the tracer, which we inject into the patient, and then get the signal, which we can measure to get some outputs, measurable outputs, which are important for us to understand the disease. And there are many aspects there which we have to cover, whoever is involved in all those stages of imaging. So these are standardization, harmonization, reliability or repeatability, which are very closely related, reproducibility, and then how that feeds into more efficient clinical trials. So when we talk about standardization, that's what we actually would prefer to have in any imaging or any medical measurement. That is, everything is standardized, everybody does the same thing, everybody uses the same equipment and calibrated to the same standards. But everybody knows it's impossible because, first of all, this world is quite free, and that's very good. Maybe in totalitarian states that is easy to do, but as you see, different sites have their own scanners, and sometimes they are purchased at different times. So some technologies are newer than others, or there are different vendors used. So the measurement will not be standard in that respect. So what then we have to do, knowing that the measurements come from different devices and performed by different teams, we need to harmonize them. And of course, many of these, like image acquisition and image reconstruction, so there will be hardware and software involved in that. They will use different techniques, and usually those techniques are protected by intellectual property, or they were very vendor specific. So they really care about, and they keep it secret, obviously. But then we use it and we need to make sure that we're getting the same answer. And then image analysis and data analysis. I think everyone is going through the pain of data analytics done with different software packages, and then you get different answers, and you pray you will be somewhere close where you are in the literature. Or maybe you are better, but you don't know. So understanding what's going on behind the data, how it's being generated, is of particular value to us, and that's what we want to do. So it gets even more complex when we have to do that across different centers longitudinally. So you repeat that, these two pipelines, and what can sometimes happen, I would imagine that for those who run clinical trials to have greater flexibility, something may happen in one site all of a sudden is not available. How about using another site longitudinally? You would rather prefer not to do that. You prefer to do that on the same site with the same people for the same participant. But we, in our DPUK network, we looked at it, what would happen if we actually use different sites. And actually results are promising. So when it comes to the efficiency, I will just focus on a few bits here. As Donna already mentioned that what is very important for clinical trials to ask the right question at the right time with the right trial. So that is one of the first steps to do. But from imaging perspective, what I would like to focus on mostly is on the uniformity of protocols. That means very much standardized, and then going into the quality of the signal which we can detect, because that will lead to novel biomarkers. So now a little bit less text, a little bit less talking text-wise, but now just looking at images. So I would like to just bring it a little bit closer to you, what it means imaging and what problems we meet when analyzing data for clinical trials. So this is just one very simple, without any quantitative corrections, PET image. And this is just one realization. Imagine now that you could do the scan again, exactly the same position, exactly the same injection of the radio tracer, and we can simulate that. So what you would do, you would see is different noise realization. I hope you can see that on the image. But then what happens? You want to analyze those images by allowing MR data, like T1 weighted data, to come in to help you with the analysis of different brain regions. And this is a segmented T1 weighted image. But when you co-register that one to the PET, different realizations, you will see a jitter. And that is one of the reasons why analytics can go wrong, or adding additional noise to the signal which is already there. Okay, so now when we look at the map, so this is just a gray matter map of one of the image slices. The blue area corresponds to the common areas as they are segmented for gray matter for both noisy realizations, and the white and red areas are separate. If we just look at gray matter as a global thing, we usually get that average out and we are quite happy. But with time, we have to look more accurately in different brain regions. And then when we look at this like singular gyrus, different parts, we may be very much off. And that may actually explain why some analytics are producing very variable results. So what we do some studies to further understand that, we simulate the data. I mean, we go really deeply into the acquisition of every single photon in the scanner, we resample that and to generate different images and see how analytics will perform. So here we have different registration software. We look at different amyloid beta positive and negative late and early time frames and different reconstruction types, whether iterations or reconstruction type, quantitative and non-quantitative. And then we see the darker boxes mean that the jitter was minimal, and those dots correspond to those registrations which perform statistically equivalently and also very well. Another thing that's very important, because as you know, any imaging has limited resolution, whether that would be MR or would be PET. PET usually, it's, in terms of resolution, is a little bit lower, generally speaking, than a structural MR. But we can do, with the help of MR, because we know where grey and white matter is, we can do partial volume correction. So you can see at the top row, this is just negative amyloid scan. And on the right, you can see partial volume corrected. So you can see the which is essential for reading those scans, the contrast between white and grey matter is enhanced. But what happens at the bottom, what we can do with our analytics, that we can see how much there is error in it. That means the uncertainty for every single voxel. And when we move it to the partial volume correction, you can see that the error, or the standard error, is increased. And now the question is, how much error do I introduce relative to the gain signal? And that's always the question. So you can see there's big controversy, we're using PET with partial volume correction or without. And you will see people on both sides fighting who has right. But what matters is that with understanding the data, we can say when it may be beneficial and when actually you will introduce more noise. It depends which part of the brain you were analyzing, and sometimes you may average it out and you will be the net benefit for using partial volume correction, but it's not always true. Now, another thing is using phantom data. I don't want to focus too much on that. However, this is ACR says American Radiologist College, I think, approved phantom. It's also called JSHOC phantom. Sometimes it's very popular to use the Hoffman phantom, which looks like the brain, if some of you are familiar with that. But here what we look is we have a background, so it simulates, like, let's say, grey matter, and then we look at different spots which would simulate, depends, grey matter, well, different parts of it, whether it be FDG, or what really matters is the contrast between the background and a hot spot. And that might be the contrast between grey and white matter in amyloid, or that might be tau, when there's activated, when the protein in different parts of the brain. And when you see those graphs, usually this is the red area corresponds to the hot spot, the hot insert, and then the white area is the background, and then the insert wall is the plastic thing which separates the two regions in the phantom. And as you see that for any measurement, we want always a step function. So the activity is there and then it drops to the background. But it doesn't happen. There's always a loss of resolution. And what we can do is to measure that very accurately through proper sampling of those different inserts or different regions. And by sampling that, we can fit error function to it. So from hot, we're going gradually to the background, or from cold areas to the background. And then when we differentiate that, which essentially get a Gaussian, which represents our image resolution. And this is a very important thing to realize about PET imaging. The effective image resolution does change on what we are imaging. Not only what we are imaging and what kind of distributions we are imaging, but also in the part of the scanner relative to the center of the scanner. The resolution will change. And that will have always, we sometimes say, oh, the scanner resolution is 5 millimeters. Generally speaking, only because when we move to different parts of the distribution, as you can see, the Gaussians, the width of that which defines the resolution, can be different. Now, I would like to talk a little bit more about the Dementia Platform UK network. And originally, we had 7 sites, then added 1 site, which was in Sheffield, but then Newcastle is at the moment off, so we back to 7 sites. Those 7 sites, we have 3 Siemens Biograph MMR PET-MR scanners, which is a little bit older generation compared to the other 5, which have GE Signa PET-MR scanners, which also uses time of flight, albeit at the moment now slower than the most recent scanners. However, recently there was proposed, well, it's soon to be commercialized, I believe, Siemens Biograph 1, which is the next generation PET-MR scanner. So we hope at some point to get that one way or another. But what I'm going to talk about specifically now is one of the recently finished partnership grants led by Karl Herhold, and one of the tasks was harmonization. And what we wanted to harmonize is the image protocols between those scanners. And we did it with phantoms, as I already presented, and using clinical study. That's what we're focusing right now. And we had 45 healthy elderly participants in that study. Most of them finished despite COVID. So the primary objective is to quantify the measurement variability for each site. So we would look at reproducibility, but also there were some participants, roughly even more than 50%, if I remember rightly, who were traveling. So they could be scanned in Manchester, and the next time they were scanned in Cambridge or in Imperial. So what happens in that test retest scan? We have 1 point, measurement point, which will have inherent variability. In that variability, which I would like to stress here, is not just the variability coming from the network, all the engineering, all the equipment with that, all the imaging protocols, but also biological variability. So for example, if there was the question, if someone drinks a coffee or just before the scan, is that going to affect it or not? Or whether they take drugs, is that going to affect the scan or not? Usually amyloid is quite stable, but other traces like FDG, we know it, not so good. So sometimes, even if some people would be scanned with open eyes, some without open eyes, and you would be able to see that in the occipital lobe. So especially this functional imaging, we need to be very careful about. But after 4 weeks, roughly speaking, we were scanning. COVID didn't help us in this respect, but there will be some delta, and we hope that will be within some acceptable measurement error. So what did the clinical study now, because it was already presented, but just in 2023, but I would like to repeat it. So we scanned it from post-injection 60 minutes to 120. So for 1 hour, we had a scan. Within that 1 hour, simultaneously, to PET, we had advanced and standard MR images acquired. But for the analytics, for PET, we only use 90 to 110 minutes, the standard 20-minute window. And also the image data were visually read by 3 raters. So what we acquired was 77 out of 90 intended PET-MR scans. So, and that meant that 37 out of 45 participants completed both test retest data. There were some participants, 2 I believe or something like that, didn't finish. So they started, did the first scan, but didn't complete with the retest scan. So this is the example. So that's in Cambridge, one of the amyloid positive scans, time point 0, time point 1, so test retest, and another one which is Newcastle, and then repeated in Edinburgh so this is GE Signa scanner and Siemens MMR. So they looked quite comparable, one could say, which is not bad for different technologies. It can be way worse. But I would like to focus on the result, and that's the main result for that study, that all the test retest scans, apart from one, in Cambridge one, which I'm just coming back to it in a moment, they are nicely lying down on the line, which means that on the PET-MR, we can do as good measurement of PET, amyloid PET, as on PET-CT. Because on the PET-MR, there's always the problem you might have had of attenuation correction, because CT provides better estimation of the attenuating medium than MR. But even with that not being ideal, we can still achieve the same accuracy. So when we look at the Bolt-Altman plot, we can see that the test retest measurement is within plus minus 10 centiloids. That is very well within what is expected for PET-CT studies. So the good news is we can use our network with PET-MR scanners in a harmonized way, and we can deliver the clinical trials even when they are run on different scanners. This is the reason why one of the participant scans fail, is just because we use a head coil which is not 100% compatible with GE, and the method just didn't work out. And we could clearly detect that, and it's very easily fixed. So that data point can be easily fixed. Now, I just briefly want to mention that all those analytics were performed by ourselves, and with a software which is now open source, and we can do essentially any brain scan. So that was first started with amyloid, and as you can see, because sometimes we do, for research specifically, dynamic scans. And in dynamic scanning, when you have in total 120 minutes, it's almost impossible to avoid motion. Even when somebody is super still, they will always move 1 or 2 millimeters over the time. And of course, this is in the images exaggerated for the purpose of illustration, but we need to bring them together aligned before any analytics. So motion is one of the biggest killers of high statistical power in clinical trials. That's how we very easily deteriorate the signal. But then, because we need to use centiloid scales for different traces, because in our study we were using flutamethimol and florbetaben coming from different vendors of the radiopharmaceuticals, and they behave kinetically differently. So we need to use some way of harmonizing that so that the centiloid scale is used for that. And the beauty of that software is that whichever way you choose your analytics, you may choose it on a 1 millimeter voxel grid, on 2 millimeter voxel grid. You may choose your own registration tool, or you may use your own regions. Automatically, we can update that and calibrate the whole pipeline specifically for that use. And then this is another important factor, which is when we want to have very high precision and accuracy clinical trial, we need to have very good quality control of the imaging which we produce and all the analytics in the end. So that is for one of the scans. We can see the the centroid mask being aligned. But if we want to go more accurate and look at specifically for every participant, use their gray matter mask, we can do that as well. But mind you, if we do that, it's already proven, if we want to be more accurate spatially, we usually produce more variability in that final statistic. Unless we really understand what the variability brings in, that will usually, if we don't, it's better to actually use a little bit wider regions of interest. And in the end, what we really intend to do in the end, which is quite novel worldwide, is when we have a clinical trial network, I mean, we're talking about imaging, what we want to do, so every site has its own scanner, and every scanner is different, even when it's coming from the same manufacturer. What we want to do is there are new initiatives like for raw data. So the raw data as it's acquired from the scanner, and I'm not talking about imaging raw data, it's the raw data before even the images are generated. Whether that is MR or PET, we standardize that. And there are already initiatives for that. Not only for clinical trials, but also for AI. It's the same problem. Everybody knows that every single clinical image, whether for the trial or study, is worth gold, essentially. It's not that, other people working on Facebook images or from Instagram, when there are millions of images almost freely available for those companies. For us, it's much more difficult to obtain those images. So we cannot throw images into the analytics as others, all other researchers can do. So for us, data is paramount. It's of incredible value. So we need to treat that, with utmost care. That's why when we acquire the data, we look at that in a global way. That is, it's reconstructed to the same grid, is dealt with the same way, so that we can produce results which are then comparable, and for every time point, having an uncertainty estimation, and then feeding that further to the analytics. And that is the next way to look at imaging for clinical trials. That's before we actually address biological variability, which is totally different kettle of fish and which much more difficult to deal with. But this is the first step to deliver higher efficacy and from this technical standpoint. So on this, I would like to finish. I hope I didn't take too much time. Thank you.
I remain very impressed by correlation of 0.97 to 0.98. I mean, it's just incredible. But you did talk about the challenges. And I wonder, just for, if you like, making multi-center trials practically possible, or feasible, I should say, is there some way of focusing the biological question sufficiently precisely so that you can focus the imaging question, therefore you're reducing unwanted, even though it might be wonderfully academically interesting, you're reducing the unwanted variability, therefore reducing the time required in the machine and the distribution around the particular, whatever outcome you're looking at.
Absolutely. This is the next way of running any clinical imaging studies, for my belief, is to be precise on what we want to measure. So the biological question is, from my perspective, understanding paramount. That's why I'm very keen to talk to clinicians and people who work even on very much preclinical aspects before we get into the imaging, to further understand the signal. Because when I understand the signal, I can optimize the whole pipeline specifically for that. So what vendors do, scanner vendors, they do that, all the optimization for an average performance. Usually that performance is driven by oncology because that's where the sales are going that way. But that might not be for brain imaging. Of course, there are different things to play with, but the more information we have about the target, I can, in my last image, we lost it now, but I had all those knobs which we can change and optimize that for our own imaging trial. Whether that will be tau or amyloid, they will have different optimization parameters for best performance for any given clinical trial. So absolutely, that having the knowledge of what we're measuring, and even better to have any study, like pilot study, which we can further optimize that's luxury.
So DPUK, helped leverage the money for the original network and your harmonization study. I wonder whether we could do something similar for, if you like, tissue-specific or mechanism-specific studies, because it strikes me that would make multi-center imaging much more straightforward.
Absolutely. This is another dream. If I could have access to all this microscopy data, people working on tissue samples, and if I could relate that to imaging, then that's what I mentioned about the biological variability. We would have a way better understanding of that biological variability. Not 100%, but we would get that closer. And that would facilitate much more streamlined processing down the line. So again, I believe that is the way to go with clinical trials. So essentially getting the data from multiple avenues, if you like, and combining that in one big hub, and then run analytics on that.
Hi, thank you for your nice talk. I wanted to ask you about the qMRI. We know that getting the MRI is very sensitive, that if the patient moves or anything happens, data gets lots of noise. So why we are not using qMRI, or even in the researches I have seen, most of the people use MRI. So what's the issues with qMRI, or why we are not using that?
What do you mean by qMRI?
Quantitative MRI.
Yeah.
I mean, where we get exactly the values. In MRI, we get the values from the images, right?
Yeah.
But in qMRI, we get the direct values from the machine.
Yeah, but for this particular study, we were interested, were the different regions and how that was very much interesting, like quantitative, whether there would be volumetric data or whichever quantitative measure you're looking at. But when it's helping PET, it has what is the most important information for us is the tissue type and where precisely it is. So that's what we only care about. And of course, the other markers coming from MRI, that are valuable on its own. And that creates additional data to the PET data. So there are different uses of MRI to help PET analytics. And then another one, which is a more specific biomarker, which then feeds into the global understanding of what's going on in the brain. So there are two different aspects. So I wouldn't put them all together. So that's why MR usage for the purposes of PET is one thing, and MR usage for its own biomarkers, it's on its own. That's why we use so many advanced MR sequences on its own, and they will provide their own biomarkers.
Thank you. Pawel, I wonder if I could just ask, because you touched on it in the latter part of your talk, there's a very difficult trade-off researchers and trialists face. As you want to try and get greater precision and anatomical knowledge, you may introduce more noise, lose power. What are the principles by which researchers or trialists can make the optimal choice, finding that best balance?
Yeah, so finding the optimum is to have the right tool for the analysis, essentially. And one of the key aspects is detection of motion or of any problem automatically, preferably. So if you run, so that's what we've done at UCL, 1,000 scans. To do that manually, visually, is too daunting. So we need to have automated tools as much as possible. And always there is some manual QCing needed, but which is very much streamlined. So that is one way. And then ensuring what tools are the best for correcting the data. So that's how is the best to improve the precision in cases where things go not to plan. Because it's very difficult, I have to say, to get ideal data point, which is patient didn't move, everything went smoothly, tracer production was great, they got injected exactly as we wanted. Those days are few here in between. So you always get something which didn't work, or sometimes like the time window was a little bit off, very often. So that's one thing. Another thing is that the data will coming from different scanners, and now, which I perhaps didn't emphasize enough. We can't do much if a scanner is like 10 years old versus recent current scanner. We know that the data, the signal to noise will be better. But in current harmonization techniques, it's usually that the least performing scanner dictates the quality of the overall trial. So everything is brought down. Even if you have an amazing scanner, it will be brought down to the level of the least performing scanner in order to harmonize that. Our approach is different. To correct all the data as much as we can, and then on top of that, estimate the uncertainty coming from any measurement. So we know that the lower performing scanner will have higher uncertainty. So if we pair those values of the point estimate of any scan, PET scan, or MR for that matter, and if we are able to estimate the uncertainty, we then take that and feed that to the novel analytics, and we will know that the better scan will have more to say for the analytics, will have higher weight than the scanner, which is a little bit older, or there was, for example, greater motion, which we could not fully correct for, because there are always limitations. But the key aspect to precision is knowledge. So knowledge, everything about the data, because we know how much we can trust that particular data point.
Fantastic. Thank you very much indeed. Thank you very much for those insights. Once again, the audience, thank you very much, Pawel.
So I'm going to start just by taking you on a bit of an emotional roller coaster, if I may, after a coffee break. So hopefully this will warm you up for the rest of the afternoon. And I'm going to start just by this rather depressing graph, which was published in a paper by Jeffrey Cummings, which was published, I think, in 2022. And it was showing basically the amount of R&D spend by pharma companies relative to the approval of drugs for AD. So this was actually quite sad. But in fact, it wasn't as if nothing was happening during this period. And, I think arguably that R&D spend has now been shown to be, quite successful. Because in the background, there was an awful lot of work done in terms of characterizing the disease, developing biomarkers, and really starting to understand the mechanisms of Alzheimer's disease, which arguably have been instrumental in the approval of drugs which target amyloid. Both in terms of clinical trial design, but also in terms of using biomarkers potentially as outcome measures in clinical trials. So then this happened, which was quite exciting. You can see the kind of slightly feverish headlines from the UK press when, drugs which show promise in terms of targeting, the processes underlying Alzheimer's disease, were first published. Of course, the Daily Star outdid itself with a slightly different take, Nerds versus Aliens, but, you know, I think that was really the outlier in terms of the excitement that was generated by these revolutionary drugs, arguably. Now, of course, the gap between the hype and the reality of this, especially when it comes to implementing it in clinical practice, is not insignificant. So these are just some data showing within the UK, within England specifically, where we stand in terms of readiness to implement these disease-modifying drugs. So although we know that obviously, and I don't need to tell anybody in this room, that dementia is incredibly prevalent, diagnosis rates are still pretty poor. So something like 2/3 of patients in England are undiagnosed. And in terms of accessing these disease-modifying treatments, in addition to the gap from a regulatory point of view, there's a real feeling that the healthcare system is not ready yet. So just over 1/3 of old age psychiatrists believe that they were ready to implement these drugs in their clinical practice within a year. The other gap that exists is in terms of the infrastructure, which is available to do that kind of precision diagnosis, identifying patients with amyloid in their brain, and the projected need for this. So currently, the UK stands near the bottom of the G7 countries in terms of availability of both PET scanners, which are the most readily available technology for identifying amyloid in the brain, and dementia specialists. So there's this big unmet need in terms of the availability to diagnose Alzheimer's disease and tie it into a specific underlying pathology. And this gap in terms of capacity, if the current standards of care continue, is going to result in wait times of up to 120 months, by which point actually the window in which patients can be treated with disease-modifying drugs could very well be long gone. The gap is also financially not inconsiderable. So it would take 14 billion over the next 10 years to bring the UK up to the middle of the G7 countries, not even kind of at a leading position. So where does kind of cognitive assessment sit within this landscape? And this is a landscape where we're seeing a lot of things like blood biomarkers emerging, disease-modifying treatments emerging. And also, where does digital cognitive testing fit into this? Isn't it enough just to do the kind of the MMSE? And I think actually there's an increased recognition that no, the kind of the standard tools that we have in clinical practice are really not going to be fit for the world as it's moving on. So even in the American Academy of Clinical Neuropsychology, and anybody who's dealt with neuropsychologists known that they're very wedded to their established tools, has pointed out that given kind of demographic shifts, particularly in people with English as a second language, or people who are coming to the US from outside of the US, although obviously that's likely to change at the moment, they anticipate that actually there's going to be 1 in 3 Americans who will not be accessible using the tools that exist because they were designed back in the 1940s, back in the 1950s, when there was a much more homogeneous population, and they were designed for essentially white European English-speaking people. So the next thing to say, and it's quite a complicated diagram, so I'll try and paint a picture with words, is that there has been an effort to think about how we can actually bridge those translational gaps and bring technologies that aren't PET scanners, that aren't pencil and paper neuropsychological assessments to bear in terms of actually making health systems ready for the emergence of disease-modifying drugs. So the way that this has been conceptualized is a really nice paper by Oscar Hansen, and you'll have to Google it yourselves because the reference isn't at the bottom of the slides, really painted a picture of a world where blood biomarkers could be used in patients with cognitive impairment who would then receive basically one of 3 diagnoses. And I think we saw this earlier in one of the talks. Either they would be biomarker positive, or they would be biomarker negative. Oh, look, we have slides back again. Or they would be in this intermediate phase, which, depending on the particular assay, could be up to a third of cases. Those intermediate patients and the patients with would then go through to kind of more expensive, if you want, imaging modalities that would give certainty as to the presence of amyloid. And then those patients would feed into to treatments. Now, if you're going to identify patients with cognitive impairment at a kind of a sufficiently large scale to address this unmet need, I would argue that actually what we need are tools which have that element of scalability that can be deployed remotely without people having to come into the clinic, and can provide information to patients and to caregivers about the status of their cognitive health, if you want, without adding additional burden into the health system. I think there's another potential role for digital cognitive assessments at the other end of the spectrum. So we know that there are these patients which are either showing negative on blood biomarkers or are showing in that intermediate range and don't meet the criteria on PET. But that's a static assessment at a single time point. What happens to them after that? How are they going to be monitored? How are you going to detect changes in their clinical state so that you can then intervene and reassess their amyloid status as the disease progresses? So this is another use case for particularly remote cognitive assessments. And finally, I think in the middle of this, it's a question, and I think there are projects which are starting to address this, as to whether the accuracy of those blood-based biomarkers can be refined on the basis of cognitive assessments. So trying to reduce that ambiguity in terms of that intermediate state on blood biomarkers. So what does kind of good look like in terms of digital cognitive assessments? So thinking about these kind of use cases, I think there are kind of three broad buckets that we need to think about. The first one is scalability, and I think scalability is a word a bit like biomarkers, which is thrown about a bit loosey-goosey. What do we mean by scalable? And here there are two aspects to this. One is technological. So is this a technology which can be deployed robustly and at scale on devices that actually make sense to people? And the second one is actually, is this bearable by people, particularly older adults who may not quite have the familiarity with technology that younger people do? So is this something that they will do and that they will do repeatedly? Sensitivity, I think we've heard a lot of sensitivity and specificity talked about. And here, again, depending on the context of use, we're thinking about sensitivity to disease, sensitivity to treatment effects, and sensitivity to disease progression. And depending on where you're sitting in terms of those different use cases, it may very well be different things which are useful at different, or should be prioritized at different stages of the deployment of these tools. And finally, the last one I think is meaningful. So are these assessments which make sense to clinicians, which make sense to patients, which are addressing symptoms which matter to them? And here there's been some really nice work, which I'm not going to go into, by the Digital Medicine Society in the US, which has looked at essentially which measures matter to patients. And the top two are really remembering where things are and language, so being able to express yourself and make your needs understood. So in terms of that first one, scalability, I'm going to talk through just an example of how these technologies can be scalable and they can address some of these gaps in terms of access to cognitive assessment. So this is a study which was run by Biogen alongside with Apple. And this was really aiming to evaluate the feasibility of using digital technologies in a large sample size. It was an entirely remote study. And the participants in this study were asked to do monthly cognitive assessment battery using the Cantab assessments. As well as every quarter, they were asked to do a burst of high-frequency testing, so being assessed every day for, I think, 2 weeks, using 2 different cognitive assessments. The recruitment on this study was pretty impressive. Over the course of 18 months, they were able to recruit 23,004 participants. The other thing which is impressive about this is not just the sample size, but also the access to underrepresented populations. In the US, when you look at recruitment into clinical trials, these tend to be clustered around, urban areas and areas where there are, tertiary centers. What they were able to do was to really recruit a very broadly representative sample geographically with the New York and Massachusetts showing a 2.3 or a 3% oversampling relative to their population, and Texas showing a 1.2 underrepresentation relative to their population. They also managed to recruit a large number of underrepresented populations. So these are people of African American heritage and also people of Hispanic and other heritage, which is, if you look at the recruitment rates for clinical trials in the Alzheimer's space, this is impressive, although I think it's not quite there yet. And in terms of the bearability of these assessments for participants, so these are looking really at the attrition over the course of 12 months. And the three panels are for passive data collected on the Apple Watch, the monthly cognitive assessments, and then the high-frequency testing. So that was done quarterly. And one of the really interesting things from this study is that actually the group that had the highest levels of adherence were actually the older adults. Older adults are shown in orange at the top, and they're the ones with the highest rates of adherence. The ones in pink at the bottom are young participants. So these are people, in their 20s to their 40s who self-report as having mild cognitive impairment. And you can make of that what you wish. The other thing that is quite interesting is if you compare the plots across the passive data collection, so this is data collection on just smartwatches, compared to the active assessments, it's pretty comparable, actually. So we always think of passive data as being the one which is going to give us the greatest coverage, the most amount of data. But actually, passive is not necessarily always as passive as we think it is. So what did the data look like from this sample? So I'm just going to give you two snapshots. This is very kind of early days for this data set. It's incredibly rich, but this is just showing a couple of data points. So in the figure on the left, you can see kind of the expected age-related decline in cognition. You can also see the overrepresentation. So they oversampled for people that were aged 50 plus, which is that density plot. And they also used that data from the baseline assessments only to look at whether you could identify patients who had older adults who were having mild cognitive impairment. And this mild cognitive impairment, it's a bit of a mixed bag because some of it was based on telemedicine visits, some of it was based on in-person confirmation. So it's a bit of a mixed bag. So this is great in terms of scalability. I also want to touch on a different method for collecting digital data that I think has a lot of relevance for MCI and neurodegenerative diseases actually more broadly. And that's the assessment of speech and language. And now typically, or in the past, this has been quite a time-consuming effort and it's been quite subjective. But actually using digital recordings and a lot of signal processing and natural language processing, we can start to get really objective measures of speech and language and changes in speech biomarkers. So the way that it works is essentially recordings can either be captured in a dedicated device, so a smartphone or a tablet, or recordings can also be taken from other sources. So either existing recordings of clinical assessments or telemedicine visits, really quite diverse. That then goes through a processing pipeline, so looking at both extracting the acoustic features, so the characteristics of the voice, as well as speech and language features, which relate to how people speak. So what kind of nouns do they use? Do they use kind of complex vocabulary as they speak, or is that simplified? And also things like pauses in speech are something that would categorize under that. And that actually gives you a really rich data set. So in our processing pipeline, we're generating about 700 features across both acoustic and linguistic properties. So what is this kind of thing good for? So this is an example of data taken from a clinical trial. So this was reuse of recordings which were captured as part of the standard clinical assessments that were conducted in this clinical trial. And this is just data comparing the speech features and the changes in these kind of composite speech measures alongside the kind of standard outcome measures which are captured in clinical trials. So what we can see on the left-hand side is the correlations between the digital speech measures and both standard outcome measures and manually acquired speech measures, which is pretty reasonable. What is, I think, more interesting is actually capturing change over time. So this was a clinical trial that didn't show a significant benefit of the drug, but you can see that actually across all of these different measures, you get a worsening of performance over 18 months of the study visit. The speech composite is shown in the kind of orangey red color, and you can see that the rate of progression of changes in speech is actually comparable to the changes in the other more conventional measures. There's also data, and this is where I think it starts to get interesting, and I think this is where we're really kind of starting to move towards actually getting the best out of blood biomarkers and all they have to offer, as well as digital measures. So this is showing the relationship between these speech measures and measures of cortical grey matter using PET cunning. So this is tau actually on PET, which is quite nice. So you can see that there's an association there, and this is just cross-sectional. So just to kind of finish up, I'm kind of going to think, are we closing the gap? Are we starting to bridge that void between really exciting drugs that are available, or really exciting treatments that are available, and the capacity to really identify patients and monitor them in practice? And I think that, yes, to some extent. So we have amazing work in blood-based biomarkers, and we heard some of that, earlier on. So this is a field that is really developing and starting to show a huge amount of promise in terms of increasing our ability to identify the underlying pathology in patients with dementia. I think that digital biomarkers and digital cognitive assessments are also really showing promise in terms of scalability and suitability across a range of different applications. And I think the availability of things like measures of speech and language actually gives us a window into very ecologically relevant changes in people's behavior and in people's cognitive abilities as the disease progresses. But I think there are also some important remaining gaps. And here I'm not going to go into depth in this because I think it's actually something that would take a whole hour to talk about properly. But there are also gaps in terms of implementation. So the nice diagram that I showed at the beginning is entirely hypothetical. There is actually a huge diversity in terms of healthcare systems, care pathways within those healthcare systems, and also variance in terms of which blood biomarkers, which facilities to evaluate those are going to be available in those different contexts. we're not the only company, sad to say, that is developing digital cognitive assessments and looking at how these can be scalable. So there's going to be diversity in terms of which digital technologies are going to be used. And where the kind of the theory meets the practice is this idea of implementation science. So really starting to evaluate these different technologies in different healthcare contexts and not only understand the technical and scientific performance of them, but also understand how fit for use they are in that context of use, so in different clinical pathways. And this is a project which started last year. I think the anniversary is probably in July. And this is the AD Riddle project, which is led by Markiewicz. And it's really looking in the European context at implementing these different modalities in different healthcare systems and really starting to get a good feel for how they can be implemented in practice. And with that, I'm going to just thank you very much.
Hi, Francesca. Thank you for your talk. Can I just ask, the digital cognitive tests that you're talking about, are they freely available for researchers to use?
No, but you can talk to my colleague Ross over there. So, yes, you talk to my colleague Ross. I'm not allowed to talk about these things because, yeah.
Okay, thank you.
Thanks, Francesca. So you're talking about implementation science, but aren't we still far away from validating some of these approaches? What are your thoughts?
Yeah, I think if you think about validation, it's kind of, again, it's one of those words which people keep on using, and it can mean a number of different things. So I think that there are still some work to do in terms of validation from a technical point of view, which I think is important when you talk about digital technologies. Do these things work as it says on the tin? Do they work in a real world? There's also, I think, important clinical validation to do. Some of the limitations of this, if you want, is that a lot of the clinical validation that you do is in a specific context, in a particular population, and for a particular use case as well. And I think here the validation, again, goes back to that notion of context of use. Are you validating it for diagnosis? Are you validating it for prognosis? Are you validating it for monitoring? And each of those will have slightly different validation requirements. So I think validate, and that applies to both digital and to blood biomarkers. I think then there's also, and I think this applies probably a little bit more to blood biomarkers than it does to the digital assessments, but the validation is likely to be slightly different depending on, again, the context of use, depending on where it's deployed, depending on the specific infrastructure and abilities that that setting has. So I think validation is a journey rather than just a single thing that you do once and then you're validated and that's it. But I think it's also, as far as I'm concerned, it's like not letting perfect be the enemy of good. So we have things which are good. And I think that, there's such a need for doing things better that I don't think we should wait until things are perfect before we start to understand the value of them in terms of improving the status quo.
I was curious about the result that older adults were more likely to adhere to the testing. Do you think that's something that's representative, or you've managed to sample older adults who are quite techie and were enjoying doing the testing?
I think that's a really good question. I mean, I will say that across every study that we've done where we've used digital technologies in patient populations and in healthy controls, we find that generally patient populations are much more likely to actually adhere to this testing and be engaged because they have skin in the game. They really want to know how they're doing, how they're progressing. They have a real motivation to participate and to collect this data in a way that somebody who's in their 20s just doesn't. So I think that there's actually much more appetite for doing these kinds of data collections in people who feel like it's actually going to contribute to them, either short term or long term.
Hi there, thank you so much. The talk is really fascinating. My question is, given the gaps that existing cognitive assessments have in terms of assessing people who speak languages other than English, I was wondering if you have any suggestions on what can be done to prevent the same thing happening as we develop digital biomarkers and digital cognitive assessments.
Yeah, I think that's a really good question. So I'll start by saying I think the ethos I think that Cambridge Cognition has always taken has been to try to make tests as non-verbal as possible, which means that then you can really deploy the assessments across a global scale. Actually, once the instructions are translated, then the stimuli themselves are not language dependent, so that then you can kind of try to mitigate some of that barrier. I think that where I was certainly very concerned probably 5 to 10 years ago was actually in terms of the digital divide. So people in underserved communities, older adults, not having access to the same technology stack, if you want, that people with higher incomes might have access to. I think given the ubiquity of smartphones, I think that has, I'm not worried about that anymore in the same way that I was. I think one of the nice things that paper from Biogen demonstrated is that if you do outreach correctly, if you actually engage people in underserved communities, they will engage, but you have to kind of meet people where they are. And I think there's still like big gaps in that. From the speech point of view, I think that is also a really interesting one in terms of how much of the features that we extract are language specific versus language independent. Can we derive the same features across multiple different languages? Which ones are most robust to language or cultural effects? And that's kind of ongoing work that we're doing, just like one language at a time, effectively.
And final question to John.
So Francesca, thank you very much. Really, really interesting talk. And I'd like to thank Cambridge Cognition for their partnership over the years. It's been really, really helpful to us. Several years ago, I was PI for something called the Roadmap Study. And part of that was an IMI initiative. But part of it was bringing industry, regulators, and academics together. And we found that if we brought these different groups together early on, there was a really fruitful, constructive discussion. And part of that was that the regulators wanted to understand the technology. Now, I appreciate that there are several different cognitive platforms, et cetera, and that what you don't want to do is to stifle competition through over-standardizing. But to have some sort of accepted standard about what levels of validation are acceptable for which purpose would be so enormously helpful.
Yeah.
How can we achieve that?
Yeah, I mean, I think that's a very good question. And so I think that the best example of trying to do this well in a way that is actually going to, progress the field and let, a thousand flowers bloom is actually the Digital Medicine Society in the United States. So one of the things that they published was something that they called the V3 framework, which now seems to be adding Vs. But this was really thinking about validation of digital technologies really from the bench to the bedside. So thinking about how you can make sure that technologies are technically validated, are analytically validated, are clinically validated. And now they're doing some really great work in terms of patient meaningfulness. So making sure that what you build is actually going to be capturing something of how people feel and function in a way that kind of, can help. And I think, so this is kind of bridging the language of the regulators with the language of the technology developers in a way that can hopefully help to bring things together. I will say that when we think about what's been approved from a regulatory point of view as an endpoint in a clinical trial, I think that there's still, I would fair to say, a little bit of rigidity. There's been one digital endpoint approved for clinical trials. That's been in a rare disease, Duchenne muscular dystrophy, based on gait. And there, the requirement was that you would essentially, any device could do this as long as it met the same criteria for sensitivity as the device that had been used to develop that biomarker. So that's kind of an example of where we are now. I think when we think about regulatory, it's also important to think different regulators. So things like health technology, for instance, that's used in clinical practice is, again, a different thing. So here we've got things like medical device certification, software as a medical device, and that's a route that we've gone down in the past for, in fact, dementia triage in primary care and for secondary care assessment. So there are different regulators with different opinions, but I think we are starting to move towards that kind of maturity of validation for digital technologies.
Thank you.
Thank you. And please, John, I want to thank once again Francesca Cormack. Thank you.
The whole concept of the talk really is to try and explain why I'm excited about one particular type of medication, class of medication. And taking a step back, I think it's also important to consider why we should be thinking about medications that are trying to target a different mechanism of action compared to what the mainstream of the research efforts are. As you know, it's been spoken about already. We now have essentially therapies which have been shown to change the disease course in Alzheimer's disease by targeting the amyloid pathways, and I think soon we'll have results about the tau pathways as well. But importantly, I think for those of us working clinically, we know that the people who we see most of the time are people over the age of 65. And in those people, it's the rule is copathologies, right? It's very, very unusual to find people who just have amyloid pathology, followed by tau. And therefore, the concept that you can get into this group, which is where the majority of morbidity is, with a single silver bullet therapy, and then we're going to make a meaningful change in the morbidity that we see, I don't think it holds true. And that's why I've really been looking very hard at what other mechanisms lie, if you like, on the intermediary pathways across pathologies. And I'm hopeful that I'm going to convince you that one in particular is interesting to look at, and that is GLP-1 receptor agonists. GLP-1s are essentially, it's a physiological hormone. What it does in the body is that primarily it has to do with glucose. So essentially, it lowers the glucagon and it slows down gastric emptying. It also has an impact on feeding behavior. So it will increase satiation, reduce hunger, and impact cravings. Okay, so some very interesting effects. But we also know that it's got something to do with inflammation and cardiovascular function. And we're also learning that it's got something to do with the way that the renal system works and the hepatic system works. So really very broad effects. And as you can imagine, we're at a dementia conference. I'm now going to speak about what it could possibly mean for dementia. Now, taking a step back, because GLP-1s, for those of you who have, opened a web page in the past, 2 or 3 years, will be aware that that it's essentially this class of medication is all the rage due to particularly the weight loss, the weight loss indications, and you would think that this is a fairly recent thing, when actually the research on this started in around the sort of 1990s. This is one of the first studies which found that these GLP-1s, if you apply them directly into the brain of animals, you see a change of feeding behavior, in that they reduce the amount of food they took. And this was in stark contrast, because in the same experiment, when the same compound was given in the peritoneal cavity, they didn't see the same effect. So it had to do something with the central effects of GLP-1 agonists. And around the same time, they found that actually GLP-1 receptors are expressed in the brain, and actually it is found in over 50 areas of the brain, and also that evolutionary GLP-1s are pretty conservatively expressed across species. So it appears to be an important hormone for whatever reason, with a specific action in the brain in addition to the periphery. Now, I'll show you a few slides of the reason why the GLP-1s are where they are in terms of interest. This is I'm picking up one study, but it could be any one of them because obviously there's a lot of compounds. This is a study by which looked at semaglutide, which showed that essentially it's got a very strong effect on weight, and even when people stop taking the medication, although the people regain weight, it still stays off what they were treated for. So that's one of the indications for these, why people are prescribed these medications. And the other one, of course, this is primarily a diabetes medication, is that you see very strong effects on HbA1c. So for those of you who are not clinical, HbA1c is the proportion of your hemoglobin that is glycolated. So it gives you an idea of how good these patients have been in terms of maintaining healthy blood glucose levels. And also, and we'll speak about this quite a bit more, what's coming out as a class effect across different GLP-1s is a very strong effect on C-reactive protein, which is a marker of systemic inflammation. And when people looked at the broader effects of these types of drugs, again they saw something that was very interesting in that, as a class effect, GLP-1 receptor agonists have a very pronounced effect on what is called MACE, so major adverse cardiovascular events, so reduction in strokes, reductions in myocardial infarctions. And this is one particular trial, the LEADER trial, which showed a very sizable separation between the two groups in respect to these cardiovascular events, pretty much from the start of the study. Now, where does that leave us in terms of dementia? So the first things that we do is to look at pharmacoepidemiological data. These medications have been around for around 15 years now, roughly speaking, with successive generations. They're getting, gradually improved. But what we now have is access to data sets where we can look at dementia incidence. And what we see are, I would highlight two pharmacoepidemiological studies that I think are very important, and also I'll plug in our one, which just got published. The first one is this one, a paper in 2022, which compared a number of different diabetes medications in terms of dementia incidence. And what they found is that there was a very clear dementia protection effect, so reduction in dementia incidence on people in GLP-1s, as well as people who take SGLT2s. And these relationships remained even once you corrected for a number of covariates, including how well the diabetes is controlled. And that's very important, right? Because you could have just assumed that GLP-1s are better than the other antidiabetic medications in terms of how well the diabetes is controlled, and therefore all of these essentially dementia outcomes have to do simply with the better diabetes control. But that doesn't seem to be the case. Instead, when you correct for how well the diabetes is controlled, you still are getting a very clear signal in terms of diabetes, in terms of dementia. The other thing that they were able to do in a 2019 paper is to actually calculate the exposure over time of GLP-1 receptor agonists, and really ask the question: if you take this medication for longer and in higher doses, do you see higher effects? And that is what you see. So that's the bit on the left. So the longer people have been exposed to these GLP-1 analogs, the stronger the benefits in terms of prevention of dementia, which there was similar trends with metformin, similar trends with DPP4 inhibitors, but once you correct it, really it's the GLP-1s that survive this relationship. So really interesting signal. Then there was another paper which combined a large pharmacological studies in the Scandinavian states with post hoc data from 3 large GLP-1 studies. And this is the data from the post hoc analysis. So these are people who took the GLP-1s for a primary cardiovascular endpoint, and then they pulled them together and they asked the question: well, starting from the point when they were randomized, was there a difference in terms of dementia incidence? And you see that about a year and a half of treatment, after treatment has started, there's a separation between placebo and the GLP-1 receptor agonist in terms of dementia incidence. So much so that in the post hoc analysis, there was about a 30% reduction in the people who were randomized to a GLP-1. And the same paper then looked at pharmacological data from the Scandinavian countries, and they compared it against the other anti-diabetics that people were prescribed in routine medical care, and they saw that the GLP-1 was the only one that associated with significant dementia prevention. A particularly interesting question is whether there's a difference in the age at which these medications are initiated. And this study was able to look at that. This is the age at baseline. What they found that this effect was pronounced, essentially was significant in people who got prescribed the GLP-1s under the age of 70 in terms of dementia prevention. And as if you get prescribed this medication later on in life, then the protective effect becomes not significant. So there's something about timing of this effect, which I guess is relevant for us. And this is then a study that we published earlier this year in Nature Mental Health, where we did we worked with a database from the US that has over 100 million data records from an electronic health record study. It's called Trinetics. We used propensity score matching to create these different cohorts where a GLP-1 receptor agonist, in this case semaglutide, is compared against sitagliptin, empagliflozin, which is an SGLT2 inhibitor, and glipizide. And what we're looking at here is the emergence of a cognitive deficit, so first diagnosis, within 12 months of the medication being prescribed. Okay, so these are really short time periods. And what we're seeing is a very clear separation between the semaglutide and sitagliptin. Comparable results with empagliflozin, which, if you cast your mind to the pharmacological studies, it's not particularly surprising, because there is a strong SGLT2 inhibitor effect on dementia. And again, semaglutide performing considerably better to glipizide. These are very large groups of people. So we're talking about 20,000 in each group. And so we have high confidence in this data. Interestingly, the other signal that came through very clearly from this paper is an effect on addictions. Okay, so there's something that this medication does that has to do with dementia and cognition, but also in terms of dementia. Now, Novo Nordisk are the company that is actually pursuing a GLP-1 receptor in Alzheimer's disease, and these are two trials, EVOKE and EVOKE Plus. I was an investigator on those when I was at Oxford, in which they're looking at essentially either early AD or MCI, and they essentially gave people semaglutide for 156 weeks. This study is now is fully recruited, and we're going to look at a readout of this in the next, I believe, year and a half, so 2027, I believe. Don't quote me on this. Now, all of this begs the question, right? What is the potential mechanism of these drugs? Because this is very consistent data across any epidemiological study that looks at GLP-1s and dementia finds this strong signal. What could be the mechanism? An obvious mechanism is to just say, well, clearly this is cerebrovascular, because you've got all of these studies. So these are the main GLP-1 studies looking at, semaglutide, dulaglutide, liraglutide, lixisenatide, exenatide. All of them have very strong effect in terms of reducing non-fatal strokes. So what what could be assumed is actually what you're seeing is a reduction in vascular dementia. So maybe that's the whole story here, which is, you know, significant in its own right. However, there's another potential here, which may be complementary, and that is we saw the strong effect on C-reactive protein. And what we know from preclinical studies is that actually GLP-1s interact with neuroinflammation in the brain. This is one study which essentially looked at Parkinson's models in mice, where they exposed essentially a mouse model of Parkinson's that's created through the introduction of alpha-synuclein. And what they see is that the alpha-synuclein activates microglia, which in turn causes an activation in astrocytes, which are then causing, through their hyperexcited state, causes neurodegeneration. And what they did was they treated these mice for a month with a GLP-1 receptor agonist called NLY01. And what they saw was that this GLP-1 receptor agonist reduced the level of microglia activation in the brain. And also it reduced the level of downstream neurotoxicity, very pronounced effects. And then this also had behavioral effects. So the mice that were pre-treated did better cognitively. So there is definitely a story around neuroinflammation that certainly we wanted to explore. And we were able to obtain data from a trial with GLP-1 receptor agonist. This is a medication called exenatide that was trialed in an Oxford-sponsored study called Excel. 14,000 people took it for, on average 2.5 years. The primary outcome was again MACE, so looking at cardiovascular outcomes. But what they did was they looked at blood samples in 4,000 people, and they run the SomaScan assay, so high-density protein assay. And in this paper that was published last year, we were able to ask the question, okay, we know from studies in Alzheimer's disease which clusters of proteins are co-expressed in Alzheimer's disease. Are these clusters of proteins affected by treatment with exenatide? And what we found was that there was indeed an effect of exenatide on these proteins, and in particular, the proteins that have to do with inflammation. So in green, you have the baseline, and on orange, the orange is the month 12 blood test. Again, relatively short timelines as far as dementia is concerned, but a statistically significant effect on CRP, FCN2, and also this cluster of protein called M3, which is essentially cytokine interaction, which is known to be abnormal in Alzheimer's disease. So what we're seeing is a very strong neuroinflammatory signal, specifically in relation to proteins associated in Alzheimer's disease. Where that has all led me and collaborators to is to ask the question, okay, if we know that this drug has such a strong effect on dementia incidence, and we think that there is something to do with neuroinflammation, what if we gave it to people who are at risk for dementia through the fact that they have amyloid in the brain, but they don't yet have dementia? Okay, and then look at whether treatment over 12 months changes their neuroinflammatory signatures and their tau accumulation. And this is the ISUP clinical trial, which is essentially an investigator-initiated study. This is a study where you can go to in this case, an industry, to the industry, Novo Nordisk, and say, we want to look independently from you. We want to look at this research question. And we were quite pleased that they decided to go with it. So this is the impact of semaglutide in amyloid positivity trial, where we investigate the effects of oral semaglutide on tau accumulation in preclinical AD, but also we're looking at the effect on neuroinflammation by looking at TSPO PET, so looking at TSPO at a neuroinflammatory signal in PET. And then, of course, we're looking on cognition. Here we're collaborating with Francesca's team using the Cambridge Cognition battery, as well as other secondary outcomes. This is a randomized double-blind placebo-controlled study where we have 5 study sites, Oxford, Imperial, UCL, Bristol and Cambridge and Exeter. We are screening people so that we do the amyloid PET, and then we aim to randomize 88 who then get randomized to either semaglutide or placebo. It is a challenging study to run. I think it's fair to say. I see Hannah Bass smiling here, who I owe a lot to, because she's the research assistant on the study and carries this study really, because we need to be doing PET amyloid at screening, but then you have to do tau PET and TSPO PET at baseline, and then at 12 months. These are tricky tracers to work with. Very often you get scan failures and so on. The key inclusion criteria are, and that I think is a point of conversation. These are people who do not have dementia, but are amyloid positive. Okay, and so within the design of these types of studies, you need to be thinking about how exactly are you conveying this information to patients, given that essentially you're telling them at the very least they're at an acute risk of Alzheimer's disease in the next 10 years. We exclude people with diagnosis of diabetes. We allow up to 30% of people with type 2 diabetes. And a critical point here is that these people who are amyloid positive but do not have dementia are practically impossible to recruit from the NHS. So we need to be thinking about other means of recruiting them. How are we doing it? We're doing it through the research registers, such as the one that we developed in Dementias Platform UK. For example, and what we've built in is an algorithm where we recruit people on the basis of enrichment of ApoE4 carriership. So 4:1 people carriers to non-carriers are contacted. And also we are looking at a number of other risk factors together in an algorithm. We're relying on DPUK, but also other cohorts like the PROTECT cohort, the NIHR bioresource, and more recently the FAST study, in which we directly have access to p-tau217, and we're able to get that. The patients that take part of this are going through a very robust set of measures. As I said, we do the amyloid at baseline, then we do tau PET, and then we're looking at serial tau PET over 12 months, and then TSPO PET, which is the neuroinflammatory PET. The important thing to say here is that the reason why we're going for PET is because we know that people who are amyloid PET, who are amyloid positive, they have an expected trajectory of tau accumulation that is about 10 times higher in rate of accumulation of tau compared to people who are amyloid negative. And so what we're looking for here, what will be a sign that there is target engagement of the GLP-1 receptor agonist with core AD pathology, is if we see a reduction of about 25% in the rate of accumulation over 12 months. And the other things that we're doing are certainly, we want to look at blood biomarkers, and that's why we're collecting blood on 7 occasions where we can do some modeling with the blood biomarkers. Hopefully, blood that is coming out of the readout study, we'll be able to implement it into something like this, where we are trying to look for signals in a variety of biomarkers relevant both to neuroinflammation, but dementia as well. That's what I had to talk to you about. I hope that was clear, and I'm very happy to take any questions.
Just because towards the end you mentioned PET and you mentioned how difficult it is, I just wondered about it here, TSPO PET, if you remember which tracer that is, which is the second generation at least, because the first generations they were particularly difficult to deal with, and if you can also say how long you scanned the people for. If you remember that from the top of your head.
Well, I think you'll see that I'm not the imaging lead for this study very quickly. I'm relying on a collaborator at Imperial called Paul Edison, who's running with this. But we are using the tracer that's done at UCL. It's a second generation tracer. The one that, they never bother with coming up with catchy names. It's sort of numbers and letters, which sort of is where they lose me. So I guess, you're right. The first generation, really very unreliable. The second generation, much improved. Now, the issue is that obviously there is a part of the population, about 10%, who have an allele which makes the interpretation of the TSPO data difficult. And that's why one of the exclusions and one of the logistical challenges that Hannah has to grapple with is that we have to be excluding people on the basis of genotype. So in addition to all the other moving parts, we have to, in the screening, find out which, whether they are carriers of this rare allele, which would make the TSPO scan difficult to interpret. And as far as the timing of the scan, they're in there for a bit. So let that be my technical explanation. I can always look it up and get back to you.
I just had one question or a comment around this pharmacoepidemiology, which is very exciting, using existing data and repurposing existing data to generate hypotheses that you then were able to take to a formal trial. What if you just talk us through the thinking? What's the relationship in your mind between the existing data repurposing, experimental medicine, and formal trials? How can we optimize those insights?
Well, I think there's three levels to this, actually, James. So one is, certainly we now have, at least from some countries, pharmacoepidemiological data where we can trust the cognitive outcomes, right? So it's the first step in a trial, in a discovery of novel mechanisms, if you like. Certainly, the methodology that we've used in this 2025 paper, I find it's probably, the next level in terms of complexity, because it matches people according to profile. It adjusts at the point at which you create your two groups. You've accounted for a lot of the variables at that stage. So I find it's very convincing. The second line at which we probably should be looking at is trials of these compounds which have already completed and have biobanked samples. Getting into these and looking at AD biomarkers is a very neat way of actually looking for proof of concept early on, and then together, once you have these two strains together, I guess, with some preclinical data, I think you're in a very good position to go in and say, I want to repurpose this medication.
Any final questions, Ivan? If you have other questions, do please keep them. We'll have a joint panel session in a few minutes, but if no burning questions, please join me once again in thanking Ivan.
So just to give you a quick overview then, I'm going to talk about Biobank, then SAIL, then what's next. And this is a bit of an overview of, the laser doesn't work, that's fine. A bit of an overview of kind of the logistics that we were taking around it. To do a drug DWAS using UK Biobank, collaborate with experts to prioritize and select drug targets, validate it in SAIL, and then essentially pull it back round again. So UK Biobank then. Biobank is great for us because we have a bunch of cardiovascular data, we have a lot of drug information, and we have over 500,000 participants. And a lot of you will be familiar with this sort of basic study to clinical drug use pipeline, and I'm sorry for what I'm going to do to it, but we really like the idea of saving time and money, and therefore repurposing drugs that might be more viable and more financially feasible than certain drugs that have recently been at the forefront of discussions. So we use this thing called Pheasant. Pheasant is free to use and open source, but hasn't been particularly heavily used. And essentially it is a phenome scan tool that allows us to also do drug-wide analyses, which is how we've used it. When we used Pheasant, we controlled for age, sex, and Townsend Deprivation Index, and we essentially put everyone in, and we put all their drug information, and we didn't really have a specific hypothesis going in, other than that we're going to see which drugs were related to dementia. So for our results then, essentially we only found one drug that was negatively associated with dementia, which you can see on the left-hand side there, hopefully. So bendraflumethiazide, it's fourth from the bottom. That's the only one that we found. The others are, so these are all the ones that are significant. The others are positively associated with dementia. Some of those you might expect, things like insulin. And I should say as well, this is in a cardiovascular subcohort because we wanted to start dealing with things like indication bias So this cardiovascular subcohort has people who have diagnosed diabetes, diagnosed vascular heart problems. Those are variables in UK Biobank. And then we also used ICD-10 codes. So it's less than the full Biobank population. It's 166,000 plus of sort of early dementia age, a lot of whom have hypertension. So yeah, we identified 24 cardiovascular medications, and as I mentioned, bendraflumethiazide is our most promising. But our p-value wasn't fantastic when it comes to using drug-wide association studies. You want to be having a threshold of something like 10 to the -8, and we were using a more traditional threshold, but because we only got one significant result and in the direction that we were looking for, we then took it to SAIL. These analyses, I should say, were done by Dr. Matt Brackersmith and Professor Valentina Escott-Price, not by myself. So they took it into a cohort of over 270,000 individuals in Wales, so in the SAIL Databank. And all these individuals were on bendraflumethiazide. They got data through dementia diagnoses from hospitals, GPs, and outpatient records, and they looked at cumulative dosage. And then they adjusted for age, sex. They used a different deprivation index just due to the differences in cohorts and various comorbidities that we addressed by looking at a subcohort. And essentially, the long and short of this scenario is that they also found a relationship with bendraflumethiazide in the direction that we hoped for, with a much better p-value than us. So I know this was a very much a speed run, but to conclude then, Biobank is rich phenotypic data for each participant. MRI data is available, which we have used, and you can come to our poster and have a look at that. It's not the most diverse of cohorts, which is a huge problem in the entire dementia field. We don't have dose response data in Biobank, but we try to target that in SAIL. And the cognitive tests in UK Biobank are sort of Biobank own brand cognitive tests, which makes any cognitive comparisons difficult. But again, we try to do some of these, and that's shown on our poster. And the power for certain analyses, so you have like combination drugs. We didn't have the power there to be able to look at those drugs in this specific cohort, or other thiazide or thiazide-like drugs. We also didn't have the power for, unfortunately. And we have those indication bias issues that we mentioned already. And our key drug finding was only bendraflumethiazide. When it comes to SAIL, there's a huge amount of data. It's incredibly rich. And there are demographic biases with certain groups underrepresented, and we have issues with real-time updating. And there is restricted access to SAIL, which is one of the bigger sort of issues when it comes to trying to look at this cohort. It's not an issue, but UK Biobank was easier for us to access, at least. And then bendraflumethiazide association was replicated, but they also found significant relationships between many other drugs and dementia in both directions. So then a quick what's next. We're doing more analyses. I've only looked at cardiovascular drugs before. This is the data I'm discussing. We're going to look at inflammation-related drugs. There's going to be more analyses in SAIL, and then validation and other data sets from DPUK. We want to apply the approach to other conditions, as I mentioned, and then the intention is to take the data into platform studies, and then also maybe something else. So as I mentioned, you can see our poster. It also includes Mendelian randomization data that was done by Dr. Fatima Kui and Professor Hugh Markiewicz, but it's not my specialism, so I haven't gone into it here. And then, yeah, just acknowledgements and thanks to all of these people. And I mentioned that I am also funded by DPUK. So yeah, thank you.
Hi everyone, my name is Aygün Badalova. Today I'm going to talk about digital neurorehabilitation in people with dementia, mainly about Gotcha trial. So I'm working with Professor Alexander Lev at UCL, and We are doing study on clinical trial on Gotcha therapy app. So first of all, I'm going to talk about proper name anomia. So we all forget names, and from time to time, it happens to all of us. But people with dementia forget the names of people they know really well. Their loved ones, their family members, like the friends that they know for more than 20 years. And it's really frustrating. It's also heartbreaking. Imagine having dementia and also forgetting your loved one's name. It's not just about losing name, it's about losing connection, relationship, and bond. And in the next slide, we are going to see one of our study participants who have dementia and also proper name anomia.
And the first things that I really noticed was I just couldn't remember names. I knew who they were, I knew their backgrounds. I have known some of them for many years, but I couldn't. It didn't matter what I did, I couldn't remember their names. If I wrote them down myself and tried to learn them, I couldn't do that. So that you feel really awful when you know people and you can't get their name.
So as you see, it's really frustrating. And then there's a question. Can people with dementia relearn names? We found a solution. Yes, people with dementia can relearn names, and forgetting names doesn't have to be forever. So what we did, we know from previous research, which had been done 20 years ago, that if people get enough practice, they can relearn names. And we did with together with families of dementia and people with dementia and their carers, we developed Gotcha app. It's personalized therapy app that's designed to help dementia people to relearn names. It is based on error-reducing learning principle, and also it has feedback inside, speech recognizer feedback. So I'm going to tell you how it works. So Gotcha, in order to use Gotcha, we asked participants, like people with dementia, to give us and provide us 6 to 10 people that they want to relearn their names, their pictures. So we get the pictures, we edit pictures and add to app. And then we also record their voice, how they pronounce these names. And then they take the tablet, our study tablet, which app is incorporated inside. So in the 6 weeks, they practice 1 phase per day for 6 weeks. And app also is based on space retrievals, which once they successfully learn names, they also get some time. It's called space retrieval. That time, app is gamified app. So there is a game they play, and then after games, they need to recall the name again, and free recall. So what we found, we successfully recruited 48 people with dementia, and today I'm going to show you only half of the data, like 20 people's data. So we found that people with dementia after 6 weeks get better on naming, and they relearn name, and 80% increase in a group level. We also did qualitative analysis. We found that there is a big satisfaction. So they improve their communication, improve their communication, and also their response improves because caregivers gave us response. And they also had an increased social interaction. So this shows that Gotcha app works, and this type of therapy works among people with dementia. So this is our half of data, statistical data, which shows 80% improved positive trend this is Taoyu analysis, and also 8 of them reached the statistical significance. And this is one of the diagram before and after 6 weeks therapy. So before 6 weeks therapy, we also do some testing, which they don't get training on names. And then after 6 weeks, which they really get training every day for 5 days, but sometimes they can do Saturday and Sunday. And after 6 weeks, they get better on trained items, trained names like faces. And now you are going to hear the same participants after 6 weeks.
I've now done the tests and I am staggered. I remember all the names. I didn't think I would remember them all, but I knew I would remember quite a lot because I'm using them now and knowing them. And quite fluently, I can say their names to other people, and I don't stumble and I haven't lost it. So actually, I'm just amazed. I can't tell you how amazed I am, and also how grateful. That is just great. Yes. Thank you very much indeed, all of you. Thank you.
So this was after 6 weeks of therapy. So in our clinical trial, we also did MAG, magnetoencephalography. We scanned their brains before 6 weeks of therapy and after 6 weeks of therapy, and to see if there is any functional changes. Mainly, we looked at the gamma band, gamma oscillatory band. And I'm going to also show you some our findings from brain data. So we haven't analyzed all data, but today I'm going to show you just 14 subjects data. We found a large cluster in left ventral temporal area, and which there was gamma reduction as a result of therapy. And there are also many studies had been done on gamma and showed that gamma reduced after learning, which is consistent with our findings. So our next step is to make Gotcha publicly available, which is going to happen next year or year after. So everyone, people who have family members who got dementia, they can access Gotcha app, they can download the app, and they can add the family member's name, the loved one's pictures, and they can get trained and relearn. Because names are not just a word, they are relationship, they are bond, and they are story. And everyone deserves to be on their story. Thank you.
I just wondered, people with without dementia but with memory, working memory problems or attentional memory problems, say in the context of anxiety, depression, or other reasons, also have sort of naming problems. Have you looked at those people as well?
We only looked at people with dementia. That's what app designed for. But if you are looking, if your question is about was the people who had like low mood score or attention, yes, we looked at that covariate as well when we were doing our screening. But our study participants only diagnosed people with dementia, either vascular type or Alzheimer type.
Thank you very much. That was very cool to see an intervention with kind of therapeutic effect there. I was wondering, did you do any kind of further follow-up testing post the 6 weeks to see whether any of those benefits continue on when they're no longer completing that therapy day to day?
Thank you very much. Very interesting and good question. Yes, we did. We randomized after 6 weeks therapy, and we randomized them to group, maintenance or non-maintenance group. So Gotcha maintenance group means like they need more top-up, and non-maintenance groups, they didn't get any therapy after 6 weeks. I didn't show the data about the maintenance, but future presentation I'm going to show. Yes, we found some like results. Yes, maintenance works. It means like if they get more practice after 3 months, they can get much better, and the effect can sustain.
Thank you. Thank you very much indeed. Please join me once again in thanking Dr. Pawel. Thank you. And as we come to the final 20 minutes of this session, I wonder if I could ask the keynote speakers, Donna Wilcock and Pawel Markiewicz and Ivan Koychev and Francesca, if she's here, to come and join me up on the stage.
So I work on a study in East London where we see a lot of patients who do not have MRI scans. They go for CT scans, and this is for a number of reasons, either metal implants in their body, they can't tolerate the claustrophobia of an MRI scanner. So we see a high number of CT scans. What do you think the value is of CT scans in research? Do you think it can be used? How can it be used best? Can we use it to make synthetic MRI scans, or do you have any other thoughts on that?
Perhaps from a technical perspective, I'll come Pawel, and then from a clinical perspective, maybe Ivan would want to talk, but Pawel, what do you make of CT?
It's definitely better than nothing to start with. Of course, the CT is not so good in differentiating soft tissues, but there is some information. So there is, I would believe there is a scope for generative AI, but it has to be done very carefully because I like very much the way AI works, but it can hallucinate a lot, and you don't want that, especially in those very important studies, if you want to do that as a study. So I'm very much a measurement guy, and if there is signal, I like to give it some help. And the good thing is that in CT there is some signal, but How much, that requires a little bit more investigation and actually exactly what you want to extract from it. Maybe on the side note, what I don't like particularly when people use structural information and they want to get functional information out of that. That's just a bit of voodoo science. And you may get some information, but it's just pushing it too much. But in this case, I would be a little bit hopeful. But again, it has to be done carefully. And another good thing is, with CT being way cheaper than any tomographic modality, you can throw a lot of numbers. When you throw a lot of numbers, that means that even if your signal is not so strong, you can get, because of the numbers, you can get something out of it. So there is a potential, but to what degree that would require to be seen. That's all for me. That's from the UK perspective.
Yeah, I mean, certainly from the psychiatrist perspective, I can tell you, James, that it's very unusual for people attending old age psychiatry clinics to get anything else than a CT, and when they get that. So I did actually, as it happens, a couple of weeks ago, I put very much the same question to an MRI company that I was talking about, where they've got this, amazing algorithm looking at microstructural change and so on, and they were asking me, which clinic should we go to first to show them these amazing things? And I was like, well, everybody uses CT. Couldn't we do something with CT? And that really threw them, right? Which surprises me, because ultimately, CT head is where the bulk of the data is. And to my mind, I'm no analyst, but it seems to me that if you can develop methods that are extracting something useful, using advanced methodology from MRIs, you should be able to get something meaningful out of the bulk of the data, which is the CT. I don't know, probably Tim has talked about this for a bit. No? Tim, do you want to say a few words?
I think I'm too addicted to MRI to answer objectively. But I think you can get structure from CT, and maybe it's better than nothing, and modern CT scanners are not bad. But I just think the multimodal nature of MRI means you can get an awful lot more from it. So yeah, and I know this is sort of converting CTs to T1 MRIs. You might as well just look at the CTs, I think, because you're not going to get any additional information from that. So
CT still have a place in the Indiana Alzheimer's Disease Research Centre?
Yeah, so no. The short answer is no. And I think just in general, we don't really see CT being ordered by neurologists when there may be a cognitive issue. The main place where we see, brain CT still being done is in the emergency room when there's a potential stroke. And actually, this has become an issue with the new therapies when it comes to ARIA, because a CT scan will not pick up ARIA. And unfortunately, the few cases of deaths that have been reported from the clinical trials were because TPA was pushed after a CT scan. And unfortunately, the patient had ARIA that wasn't detected on CT, and they ended out just with massive hemorrhages. So I'd say, if anything, in the US, now that we have therapies, there's a push even more to have MRI be the go-to.
Yeah. Thank you. And I wonder if Tim might want to come back on that.
Apologies to hog the microphone. I was going to ask a different question. If
okay. That's great.
I was going to, because in MRI, we're seeing sort of low-field, cheap MRI that might be applicable to a wide array of sort of places and contexts. Will we ever see a cheap, widely available PET scan?
Well, I guess that's for you.
You're asking too much, I think. At some point, I would believe so, if it became something made. So if we look at FDG, which is mostly for oncology, it's a fraction of a cost of compared to any other tracers we have to use. And all the logistics, it's just a nightmare if you want to deal with, that's why I was asking about your TSPO tracers, because they are amazing tracers, but to make it logistically working and reliably, that's why it becomes very expensive. But if we make it move it mainstream. That's why I think it's very important for us to work quite a lot at the beginning to make the case. Once making the case and becomes more mainstream, it becomes cheaper.
But yeah, it's a real issue in terms of, it's not so much the scanning itself, but really it's the tracers that it's complete unknown. Things change from a year to year in terms of who's the actual provider of that particular tracer in a country in terms of licensing. I was recently trying to get a quote and just to understand how much a tau PET would cost. And it turns out that was a question that was unanswerable, which I found was interesting, The admin and business people getting into the research sort of ethos of saying, well, we don't have enough data, which is great.
But some of this is scale. FDG PET 10 years ago used to be 1,500, 2,000 scan, and it dropped to around 200. And that was really on the back of oncology. It's a massive surge in demand, there's a business case made, and we still benefit as neurologists a little from that oncology gain, but it was driven by scale through another discipline. In the US, is PET pretty standard available? That you heard earlier that it can be difficult in the UK.
Yeah, so PET, now that there are treatments, insurance companies are more likely to pay for the PET scans, but it is not bringing down price at all. And the cost of the actual scan is very cheap. It's the ligand, and I would say now that there's demand, the price of the ligand's actually going up.
Okay. And given the data you presented on the correlation between the blood-based biomarkers and PET, 0.95, 0.96, 0.97, depending on your study, do we actually need PET still?
It's a wonderful question. So if you look at the appropriate use guidelines and you look at what Medicare and most insurance want when it comes to therapy, they want either a CSF biomarker or PET. They will still not accept a blood-based biomarker as confirmation of amyloid status. I think the way that a lot of us are viewing the early rollout of the blood-based biomarkers is going to be to narrow down how many people we're doing PET scans on, so that we, if they're positive on the plasma, then we can do a confirmation PET to satisfy, the payers. But I think within a year or two of this being widely used, we'll see these rules changing.
Yeah, thank you. Any other questions? Yes.
I have probably a question for Donna, because I know Donna alluded to this earlier about development in technology. I just wondered if the development with MRI, because I think we've got to Tesla 7 now, from 1 or maybe 0.5. Have they offered any new insights into finding anything else about dementia?
Yeah, so there's a few 70s. I think you're asking about the increased magnet size, and is that telling us any more information? So there are a few human 70s around the States now. I've seen a couple of papers that are giving a little bit more information about kind of white matter connectivity and regional connectivity, where the 70 and we have a small animal 9.40 in Indiana, where those are, I think, most useful now is that we're able to do really detailed scans on postmortem autopsy tissue and get some new insights and be able to, for example, when we see white matter hyperintensities on MRI, if we can do post-mortem scans on autopsy tissue and then actually dissect out the specific regions that we're interested in based on that MRI, we can do some biochemistry and try and understand the etiology of those pathologies more. I haven't seen a big leap in knowledge with the 7T. The 3T is certainly the standard that we're aiming for when it comes to detection of ARIA. In theory, you can do a 1.5T, and I'm seeing some companies now that are using 1.5T with AI technology that seems to come close to the sensitivity of 3T. But for ARIA, we're using 3T.
And from a research perspective, the UK is currently building a national research centre with 11.7 for human use. So within 2 years, possibly 3, there'll be a human 11.7, and that might give a leap forward. Some of the pathophysiological processes you can see, either imaging at 100 microns or some of the spectroscopy and chemistry, but not clinical.
Right.
It'll be a research tool. Yes. No, wait for the microphone. The acoustics here are not brilliant without the microphone. Thank you.
Hello, thank you very much for the wonderful session today. That was really, really interesting. Angelina, a PhD student based in Scotland. I mostly have a clinical question, actually. With all these disease-modifying therapies that we have right now, is there ever a worry around them extending the lifespan of people with dementia, but not the health span? And what do we need to do to prevent that?
Yeah.
I would say there's no data on lifespan right now with those drugs. And I think a big focus of the trials and a big focus of us as we implement it in the clinic is quality of life. So extending extending independent living, extending good quality of life. Lifespan is going to be data we get maybe in 10 or 15 years with these drugs.
James, if I can touch on this. I think it's a very interesting question because it went directly for the health economics of these treatments, right? As it happens, I was on the NICE committee that looked at the first one, at lecanemab, and that was a point that came up, that actually if you have a therapy which is prescribed in people who are elderly, if you increase the lifespan of people with dementia, in a way, the health economics models can do something very, very unusual, very weird, in that it can actually extending the lifespan of somebody who's assumed to have low quality of life can actually make it sort of seem as if the drug is actually causing more harm than good, which just completely destroys the health economics of it. And conversely, the other thing that sort of really struck me at that health technology appraisal was that by the same token, if you look at the time that the carers of people with dementia live with somebody with the disease, if you manage to extend their life with a drug, it somehow acts against, it's evidence against the drug, because the patients, the carers of people, their relatives will have to live with somebody with dementia for longer. So it really struck me that basically the way that we do health economic evaluation is really poorly fitted to the people who are elderly and people who have sort of a significant disease. It's works brilliantly for oncology, but I think, it made me think that NICE needs to have a think about its methodology specifically in relation to our area.
Thank you. Francesca, I had a question for you, because you talked very clearly around digital biomarkers, very well prescribed, controlled biomarkers, not biomarkers based on our digital footprint. And we sometimes hear about digital footprint, which has always worried me. My brain is similar to my brain 10 years ago. My blood is the same as 10 years ago, but my digital footprint changes year by year every time a new phone comes out. So can you clarify digital biomarkers versus digital footprint?
Yeah, I think that's really interesting, and there have been lots of people that have been trying to use digital footprints and and ways in which people interact with their phones to basically derive a digital biomarker from it. I think that To me, I think this is slightly problematic for a number of reasons. The main one being, and I think this is to your point, that it has to be referenced against what's happening in the general population in terms of the ways that they interact with technologies, new technologies becoming available, and it also needs to be benchmarked with respect to the individual themselves. So the way that I use my phone is very different from the way that my 17-year-old daughter uses her phone. Thank goodness. But so I think that there's also, thinking about changes within participants. I also don't know if patients are ready for this yet. So we're involved in another EU-funded project called IdeaFast, which is looking at fatigue. It's looking at transdiagnostic digital measures and of fatigue in patients with neurodegenerative disease and inflammatory disorders. And there's a combination of active and passive data from sensors and from phones. And one of the pieces of qualitative feedback from people was that there was a reluctance to actually install the app that allows for your tracking on their phone because they found it essentially, to paraphrase, creepy. So I think that kind of, the creepiness of things listening in on you is something to consider, even though we give away huge amounts of data all the time to large corporations and we give it away without really thinking about it. Somehow making it explicit that we're looking at your data and we're kind of like seeing how you use your phone changes people's perception of it. And I don't think we're quite ready for prime time on that yet.
Thank you. Any other questions for the floor? Yes, gentleman over there.
Hiya, thank you very much, first of all of you, for providing such amazing talks. As an early career researcher myself, it's very insightful to have these new ideas and these new inspirations for what I can achieve for my research. So thank you for that, firstly. I had a question with regards to what a patient may be expected to be experiencing over the next few years and how each of your respective areas may influence a patient's choice in terms of coming in, getting a diagnosis. Like, what will the patient pathway be looking like in the next couple of years with respect to all of your different fields, and how will it impact on their care?
Thank you. I think it's safe to say it's changing very quickly, but why don't we just work our way along the panel?
Yeah, I mean, the good news is that I think it's almost a given that within a 10-year horizon, we will see some changes. What those would be, I think, largely depend on whether we do or don't have disease-modifying treatments on, being part of the armament of what patients will get, certainly on the NHS. I think it's clear that they are already part elsewhere, but as you know, certainly the two therapies that have been put forward have not met with nice approval yet. So to me, that's the first thing, that it's a bit uncertain at the moment. But then the thing that I think we will need to, our patients will be dealing in a different way 10 years from now, is that we will need to be giving them some biomarker data. Okay. So this is a study that James and Vanessa and I are working on, specifically in terms of blood biomarkers. This is the readout study where we'll be looking at what is the utility of blood biomarkers, and importantly, what is the utility of actually disclosing the results to the patients and the clinicians, and then looking at their outcomes and so on. So the fact that we're able to run this study today and then we'll have the results in 5 years means that, we will be in some position to be talking certainly about blood biomarkers in everyday clinical practice, I would say 5 years from now. So to me, that is very important. The other thing that I find will be different, probably again comes from the disease-modifying treatments. So the two therapies that have been approved require us as clinicians to test for ApoE4, right, for genetics. That's never really happened in a regular, if you like, memory clinics. The neurologists have been doing it for a while for some conditions, but for mainstream Alzheimer's disease, it's completely unheard of. And so if we're ever thinking about these therapies, a part of our pathway needs to be actually talking about genetics, okay? Because, basically these therapies cannot be prescribed to people who are homozygotes for ApoE4. So for 10% of the population, you'd be telling them both, you can't have this therapy, as well as your immediate relatives have a 100% chance of being at least carriers, at least heterozygotes of this ApoE4 gene, which carries these and these risks. So that's the focus of another study that we're running, where we are testing what is the experience, the patient experience and the clinician experience of doing ApoE4 counseling in a memory clinic. So these are some developments which I think are unavoidable now. But there will be, the big unknown factor is, what would be the extent of access to amyloid therapies in particular. But
thank you. Yeah, Donna.
Yeah, so I can tell you kind of what the picture is in our clinic right now. We are administering lecanemab, and we just started administering donanemab as well to patients. We have about 130 patients receiving anti-amyloid therapies. We've screened like 350. So we're implementing some digital tools in primary care to screen for cognitive impairment. And then we have community health workers that follow up with anyone who tests abnormal on those cognitive assessments. They administer a MOCA, they do a medical record review, they look at blood work. Are there any specific, signals like B12 or anything in that could be driving the cognitive impairment? And then the patient is offered several options. If they are found to have a cognitive impairment, one of which is our brain health program where we're administering the drugs. And you're right, so we have a funneling process that patients go through. It starts with an MRI to rule out any excluding factors like microbleeds and microinfarcts and superficial siderosis, and then we do an amyloid status. Right now, most of that is amyloid PET. Provided they're amyloid positive, they then move on for ApoE genotyping, and once they've done the ApoE genotyping, there is a patient, caregiver, physician, social worker meeting that discloses, counsels through all of the findings, and there's a shared decision-making that happens among that group about whether to pursue the therapy or not. I will say we're not excluding 4/4s. We have a number of 4/4s that are receiving therapy successfully. So our ARIA rate is 8% across our whole clinic, and only one of those is a 4/4 homozygous.
Yeah, so I guess in the UK, it's specifically part of the label. You're not allowed to prescribe it if they're E44s at the moment. But maybe that will change with the US experience.
Yeah, I think as we get more experience and we get more data, we're going to understand more about the 4/4s. One thing we have done in our anti-amyloid clinic is we've been very, very diligent about monitoring blood pressure. There's some data just come out from Lilly that after ApoE4 status, uncontrolled hypertension is also a big risk factor for ARIA. So we now reinforce hypertension control. We take blood pressures at every infusion. If blood pressure's high that day, they don't get infused. So everybody's learning as we go with this. But I think, as the new trials read out, the GLP-1 is going to read out, the EVOKE trial and the AHEAD trial in prevention, everything is going to evolve with that. But we know we don't have enough providers, and so digital tools, the blood-based biomarkers, I think these tools are going to be necessary for us to address the need that's out there, because there just aren't enough providers.
Thank you. Perhaps I could then, just in the last 2 minutes between now and the drinks reception, are we all invited to the drinks reception? I'd like to come back down the panel and say very quickly, what are you looking forward to? What are you most excited about expecting from biomarkers in the year ahead?
Well, some kind of revolution will have to happen, especially when you look at if very poor population demographics. There are more and more aging population. It has to happen, greater breakthroughs, because otherwise this situation will become unmanageable. So I'm anticipating some more and more research going to that.
Thank you, Francesca.
I think for me, it's actually that there will be, I hope, actually just more interest and more adoption of new ways of doing things. And and I think things aren't perfect now from the point of view of biomarkers, and and we know that we've still got some way to go. But I think, actually pushing these things out, getting that validation data. And for me, it's also about that combination of different biomarkers, because I think that the more that we can knit together our understanding of all the different biological, all the different digital measures, I think that's going to be the way forward, rather than it being one ring to rule them all, because I don't think that's going to work.
Thank you.
Yeah, completely agree. I think we really need to embrace digital biomarkers. Our neuropsychologists, I know, are reluctant to let go of the pen and paper, like 5-hour testing, but that's just not feasible. So I'm really excited about digital biomarkers, and I'm really excited about the idea of getting biomarkers for those non-AD dementia-causing pathologies.
Well, James, the one that I'm most excited about is work that's ongoing in my group, looking at not just about biomarkers, but biomarkers in response to a challenge of some sort, right? So the thing that my PhD student Courtney is working on is we're giving people intranasal insulin, right? And then recording the responses of the brain to insulin using MRI in this instance, so functional MRI and ASL. And to me, that's a very exciting area of research, because once you're able to work out what is the responsiveness of the brain, in this case, to insulin, you can start asking questions about, for example, why do some people with Alzheimer's disease pathology go on to develop neurodegeneration while some others, live and die with significant neurodegeneration without developing dementia. And so I'm hoping that, at some point, we're actually going to be thinking in a dynamic way about biomarkers, where we perturbate the system and we're thinking about potentially drilling into the mechanisms that underlie the disease.
Fantastic, thank you. That's a perfect way to end the day and to segue into tomorrow's sessions around trials and treatments and interventions for dementia. So I'd like to thank all of our fantastic speakers today. Thank you.